to Vedantu NEET English once again. So we are back after a lunch break, but between that, what happened, sir?
Students, our first lecture got somehow cut in the middle because there was a power cut or internet cut in the office.
Yeah.
So the entire stream got ended. This is going to be a second stream right now, and in this stream we'll be teaching how many chapters, ma'am? Evolution,
Human health and diseases, biotechnology principle, biotechnology applications,
principles of inheritance, and ecology.
My God.
Oh my God, students.
Students, in case the important questions, in case the session goes live and we are not able to come live, don't worry. We will share the PDF on the Telegram channel, so you can solve and practice. Okay?
Okay, students. Students, quickly let us know in the chat if you are audible and visible, because very important.
Yes, very important, students. So many things are happening. Like so many technical issues, so that's why we are very tensed.
The camera will not work,
exactly.
the mic will go off. So students, let's not waste any time, we can start, ma'am.
Yes sir. So see you after this for principles. Okay? Yes students, after evolution principles, so that the entire genetics can get over at one shot. Okay? Microbes is silly chapter, students. You should be able to do microbes by yourself. Okay? We are mainly focusing on mark scoring chapters, like in the sense chapters that is difficult to understand, but at the same time it gives you enough marks. Okay, chalo, now make the session to 200 likes. Okay, if you don't have Telegram, don't worry, we will put it in the description box. Okay, we'll put the link in the description box. Done? Okay, chalo, let's start evolution. So evolution, we will start with Big Bang theory. Okay, after having lunch, little sleep, we'll be little sleepy, so we have to study something interesting, but unfortunately in NCERT difficult to find. Okay, come on. Big Bang theory. So people were like, you know what, universe was found because of a Big Bang theory. Now what is this Big Bang theory, ma'am? I only know the Big Bang theory from Netflix. Let me tell you, Big Bang theory states that there was a huge explosion, singular huge explosion. The earth was formed around 4.5 billion years. So they are telling that because of the huge explosion, yes, biology will be completed. Okay, biology will be completed. Yes. Now for sleep to go, you have to all stand up and study. Okay, come on. See students, they are telling that there was a big explosion that happened that led to expansion. Okay, explosion led to expansion. And students, when something expands, automatically cooling down will happen. So temperature went down. Okay, they are saying that the temperature went down, because of which there was formation of some gases like your H2, okay, helium, etc. Okay, so this is how universe formed. That is a Big Bang theory. A big singular huge explosion happened that led to expansion, that led to cooling down and led to the formation of, formation of NCERT, I'm saying, senior messages. Formation of your universe. Okay, Earth was formed around 4.5 billion years. Okay, let me drink some water. Do you want to do principles then? You want to do principles, but principles also, see students, every chapter will be sleepy only, as if you people are very interested to study biology. No, not like, oh my God, I want to watch a Bollywood movie or a Hollywood movie or a Tollywood movie. Everything is NCERT only, everything is content only. Even if I change the chapter, it's not like you people are going to have an adrenaline rush that, oh my God, yes, I want to study this chapter. Please students, don't play with me. I know you people from now 2 years. Okay, I know you people very well. I know till where you people will, go. Okay, I know exactly till where you people will go and what you people will study, right? So that's why let's not change the chapter. Let's stick to this chapter only. Clear? Okay. Yes, Trisha, I definitely remember you. Now, there is no atmosphere on the early earth. It's important to remember, students, there was no atmosphere in the early earth. Okay, water vapor, ammonia, CO2, NH3, these were the things that were there, and these were releasing them, like it was present on the molten mass. Okay, so there was no atmosphere on the early earth. Water vapor, methane, carbon dioxide and ammonia released from molten mass covered the surface. So we cannot imagine to live there. We cannot think of life being there, because it is so like scary situation that we cannot think that life would happen there. Clear? Okay, the UV rays, now very important, the UV rays from the sun came and it broke the water molecules into H2 and O2. In that, H2 is very light-hearted person, so H2 escaped. But O2 is not like that. O2 goes and combines with other gases to make other gases. Okay, wow, what a chemistry. Okay, this is how I would have taught you chemistry. I would have told, yes, this gas has escaped and this gas went and combined with that gas to form another gas. Okay, this is how I would have taught you chemistry if I were to, if I was You know, forced to teach chemistry. Yes. So oxygen combined with ammonia and methane to form water, carbon dioxide, etc. Clear to all of you? Now, slowly ozone layer was formed. Okay, ozone layer was formed, and since ozone layer was formed, we were like, okay, now that life can be on Earth. So life happened 4 billion years ago. So there was a gap. Okay, there was a gap of 500. So understand 4.5, 200. This is 20. Okay, this is 20 billion years ago. These three dates you have to remember. Okay, yes, life appeared almost 4 billion years ago. Clear? Yes or no? Chemistry. Yeah, pro. I am a pro. Okay, now students, some theories are there. We will just go through the most important theories that can come as a question. Theory of spontaneous generation. Let's take an example of, Bushra. Okay, I don't know if I'm pronouncing your name right. Okay, so now let's take Advika. Advika went to the lab. Okay, Advika went to the lab, and Advika had, she was wearing the lab coat the entire day. There was a sack of wheat there. Okay, Advika, obviously every human sweats. She also was sweating. So she removed the lab coat, put it on the wheat bag, and she went. Okay, she went to her hometown to enjoy vacation. After 21 days, she came back. She started noticing your Jerry, okay, mouse near it, and she was like, Eureka! Life happens when, dead and decayed substance stay together. Now, students, you tell me, do you agree with Advika? Do you agree with Advika? Because the theory says that life came out of decaying and rotting mud and straw, and there was a theory that says that on wheat bag, okay, on wheat bag they put a cloth, a dirty cloth, and after 21 days of incubation they started seeing mice around it. It is simple, no? There is food source, at the same time there is bacterial growth because of the sweat. Because of that, any organism will get attracted. So do you agree with Advika or not? Tell me. No, I don't know. I don't know any teachers, like in the sense I don't know the offline teachers. Okay. Now, students, Louis Pasteur disapproved this theory. Why? What happened, Sameer? Louis Pasteur disapproved this theory because he told that no, life cannot come from dead and decayed substance. Life has to come from, existing life. Okay, life has to come from existing life. So he showed that life did not come from killed yeast, but it came. So here he proved it with an experiment called with the sterilized flask. One flask he sterilized. He put a broth in that. He noticed that okay, there's no microorganism growth because I have covered it. The moment he opened, after a few days, he noticed microbial growth. So he was like, life comes from pre-existing life. Okay, pre-existing life. Now, biogenesis proposed by Reddy, Plansky and Louis Pasteur states that life my tongue is gone. Okay, life originates from pre-existing life. But it does not explain the origin of first life. Students understand? One person told that life comes from dead and decay, another person told that life comes from existing cells. So this was the two contradictory statements that came, and they proved it with different experiments, like using a meat, okay, one open meat they kept, obviously microorganism flies will come because of the flesh smell, okay, then he closed it and kept it, he noticed that no microorganisms or flies are coming. Then he covered it with a muslin cloth, like a transparent cloth where the fragrance can come out, like the smell can come out. He noticed that some flies were sitting on the muslin cloth. Then he told that no, life comes from pre-existing life. Okay? Yes. But no one could tell that how did the first life come? Not proven. Now, cosmic theory is a very interesting theory, students. What happened in the universe? Let's say, okay, this is the planet. They are telling that there was a spore of life or unit of life. Okay, unit of life or spore of life. This came and fell on earth, and that is how first life happened. And you will not believe that now also some of the Greek thinkers and astronomers believe that. Okay, this is how, okay, some of the people right now also believe that yes, there is some theory called as cosmoic theory or panspermia theory. Life actually came from spore of life. Okay, so cosmic theory or theory of panspermia, it states that the unit of life were transferred to different planets including the earth. Do you agree? No, ma'am. Now, theory of special creation, students, people who believed in God and worshipped God told that God created everything in a span of 7 to 8 days, and that's called wonders of life, right? So, some people believe that, okay? Now, the most important from question point of view, from CBSE point of view, from NEET point of view is your Miller's experiment. So, students, Oparin and Haldane told that life actually comes from chemical evolution. Oparin and Haldane Haldane, okay, told that life comes from chemical evolution. Ma'am, what are you saying, ma'am? If you ask me that, I'll hit you people, so don't ask that. Okay, chemical evolution. So that means from some inorganic substances, okay, with some inorganic substances, we can make organic substances. Okay, from some inorganic substances, we can make some organic substances, and that is what they told. Okay, that is what they told. So here they told that from using some inorganic substances, let's form some organic substances, and that organic substances accumulate and finally form the protocell or the procell. Okay, like what? Many amino acids together form proteins, simple as that. Now to prove this, Urey and Miller did the same setup in the laboratory. Now what is the setup, ma'am? They took the same gases that were present in the early atmosphere, that is your methane, ammonia, water and H2, and to supply the vapor they gave the boiling water. Okay, and what did this Oparin and Haldane tell? They told to the earlier there was a thunderstorm or like a big thunder, okay, thunder or lightning that happened. Now because of that the gases, okay, ended up forming some small like organic substances. Now look here, instead of that lightning, here Urey and Miller gave electrodes, and that electrodes at 800 degree, they did a spark discharge at 800 degrees Celsius, and that ultimately condensed and we got a water containing organic compounds which was rich in amino acids, Like this, students, many people did the experiment and they ended up getting fats, nitrogenous bases, sugars. So that kind of gives us the conclusion, that okay, maybe actually life happened like this, that some inorganic substances condensed with the help of some energy or lightning, and then they end up giving us some organic substances, right? So that is what here they are telling. Clear? Yes or no? Urey and Miller experiment is clear. Now here, paleontology, that is evidence of evolution. How do I say that okay, dinosaurs existed? How am I so confidently saying that dinosaurs existed? It's because of the fossils. Okay, did you all understand? Please put thumbs up in the chat box. Yes, Oparin from Russia and Haldane from England. Put thumbs up in the chat box if you understood quickly. We have so much more to go. Are you people happy that I came without mask? I'm very happy. I can finally see my face and I can show you my face. Okay, I'm very happy because 1 week was not easy. Almost I think 10 days that few classes I've taken. Chalo, very good, clear ma'am. Now paleontology means what? Fossil, students. All of you notice in small age when we used to get this flower or we used to get some leaf, we used to keep it in NCRT, we used to close it and keep it, not in NCRT, that time you didn't have NCRT, in my head only NCRT is there. We used to close it and keep it in an encyclopedia, and when we used to open, we used to get beautiful patterns. Okay, so what is that? That is basically preservation. You're preserving it for the longer. Same way fossils also, because of continuous weathering, their bones would have been in the lower, what is that? Okay, like at the different layers, okay, of the soil, these bones will be present, and when these people dig, paleontologists dig, they will find some fossil and they will try to relate it with what we have in the current. Okay, so here, understand students? Yes, thank you so much. You are also very happy, even I am very happy. So fossils are remnants of life forms found in rocks, that is in the earth's crust. So significance of paleontology is, significance of fossils is what? To study the physiology. That means we understand the evolutionary history, right? To study the connecting link, like your Archaeopteryx. That is, we understood that this Archaeopteryx is so similar to that of reptiles as well as birds. How did we understand? Because of the beaks, because of the claws. That is how we understood, right? Clear to all of you? Yes. Memories, what in paleontology? One second, sorry. Okay, second. Done. Okay, yes. carbon dating and all will come into, okay. To study about the extinct animals like your dinosaurs, to study the geological period, also to understand the different types of sedimentary rocks. Now don't have to break head for all of this, just understand significance of fossils is important for evidence of evolution. Now students, most important, homologous and analogous, I don't know how many questions come every single year from this, very important for you to learn, okay. Now students, homologous and analogous, what you have to remember? You have to remember HDAC, it's not some big trick or something, you just combine the words, okay. HDTV will always be in an AC room, okay. HDTV will always be in an AC room. So HD means H for homologous, okay, homologous, D for divergent, D for divergent, okay, A for analogous, okay, analogous, C for convergent, C for convergent. Now what is this, ma'am? What is this homologous, analogous? I'll tell you an example. Let's take an example. You are all in Vedantu NEET English, yes or no? You're all, let's say you all have common ancestors. That means let's consider Vedantu NEET English as a common ancestor. From here, next year, some of you will become doctors, okay? Saying that itself makes me so emotional, but yeah. Some of them will go to MBBS college. Some of you will go to some engineering college. Some of you will pursue some good degrees. Some of you will go for like, fashion designing or like, to abroad to continue your studies. Now, this is called as divergent evolution. That means from a common ancestors, you all emerged differently. That means even though their structure is similar, their functions are different. Did you understand? Even though their structure is similar, their functions are different. Example also you should know. Example, forelimbs, okay? Forelimbs of man and cheetah. We use it for holding, they use it for running. Forelimbs of man and cheetah. Done? Yes or no? Understood? Okay, common ancestor. That means from one point you have diverged. Now, what about convergent evolution, ma'am? So, students, here you write that they have common ancestors. They're structurally similar, but they are functionally different. Like what? Humerus, radius, ulna, carpals, metacarpals, and phalanges are same in all the organisms. But I am using it for holding the bottle. Cheetah is using it for running. Okay, someone is using it for flying. Someone is using it for swimming. So, all of them are doing different job. Right? Now, when we come to analogous, it's other way around. Even though, let's take an example, some of you might be from Tamil Nadu, some of you might be from Andhra Pradesh, some of you might be from Telangana, some of you might be from Bangalore, some of you might be from Assam, different places you people are, but all of you decided to converge at one point, okay, all of you decided to join Vedantu NEET English or Vedantu, so you have all converged to one point, right? So, this is called as convergent evolution. This is called as convergent evolution. That means even though your structures are different, that means even though the structures are different, your functions are the same. Functions are same. Ma'am, example, students, here you can remember, here you should remember bougainvillea and cucurbitaceae from plants. Here you remember potato and sweet potato. Here example, students, eyes of octopus and man. Flippers of penguin, this is the most used example. Okay, flippers, not slippers, don't write slippers of penguin and dolphin and come in the exam. They'll laugh for 2 days. Okay, dolphin and penguin. Got it? So, flippers of dolphin and penguin. How many diagrams based questions may they ask? Even I don't know, dear. I did not set the question paper. How will I know? They might ask good number of questions, they might ask like 3 of them, or maybe they'll ask 4 of them, okay, depends to them, no? They make the question paper, we just prepare you for the question paper. Done. So, this is convergent evolution. From different point, you're converging to one point, from one point, you're converging to different points. Is this clear? So, here you do not have common ancestors, understand that. No common ancestors. Done? Okay, now, adaptive radiation, again a lot of questions have come from this topic as well. Adaptive radiation is the evolution of different species. Something like, let's take an example. Okay, let's take an example. I don't know how many of you are in Bangalore and many of you might not even know the places. But students, please understand, in Bangalore, Okay, some of them will be in Whitefield, some of them will be in Koramangala, some of them will be in HSR, some of them will be in JP Nagar. Oh yeah, that's when I remember JP Nagar, we are meeting you people on March 30th. Whoever is in Bangalore, don't fail to join. The link is in the description, the first link. Just fill the Google form so that your seat will be reserved there, because we have to make the arrangement, no, for refreshment and other thing. So for that, okay, yes. So students, so here all this, okay. So what is actually happening here? Adaptive radiation means is the evolution of different species from a common ancestors. And why are they doing adaptive radiation? Because they are stuck in one geographical area. Like Australia did not have any terrestrial path, because of which they had to be stuck in one place, and that is why your marsupials have done adaptive radiations to full different types. Okay, so it is a divergent evolution. Important questions can come, is it a divergent evolution or not? Darwin's finches in Galapagos Island, initially they were seed eating, but later they became vegetarian. Some of them became what is that, students, beaks modification kept changing. Okay, beaks modification. So that is because of adaptive radiation, because they did not have any place to move further, so they changed themselves. Okay, now Australian marsupials and placental mammals in Australia, both of them did adaptive radiation, because again I told you, no terrestrial path. Okay, now here students, one important question is that even though here we are telling it's a type of divergent evolution, here you will notice that when more than, very important, when more than one adaptive radiation is appeared in an isolated geographical area, it results in convergent evolution. It results in convergent evolution. Example, Australian marsupials and placental mammals. Understood? Clear or not? Adaptive radiation means from one point we radiate and move to a different point. In Bangalore itself, one person will be from Whitefield, HSR, Koramangala. All of their food habits might be different. All of their way of talking might be different. All of their living, dressing styles will be different. What has happened? Even though they are all in a geographical area, they have migrated to different places very close by, and they have got adapted to that situation. Clear? Done and pakka. Now, natural selection is a process in which the organisms are favorable and heritable. Variations are survived. Now, students, natural selection is totally based on reproductive fitness. Please understand this. Reproductive fitness. Who is able to survive and who is able to be the strongest will reproduce and succeed. Okay. Now, here example given is industrial melanism and before industrialization and after industrialization. Nothing much is there. It's just that on trees, before industrialization, there were lichens present. Okay, before industrialization, there were lichens present, and since lichens were present, you couldn't see the white-winged moth. You could not see the white-winged moth, but all the predators would actually spot the dark-winged moth. All predators could spot the dark-winged moth. Because of which, what happened in before industrialization, dark-winged moth reduced, the number reduced, and white-winged moth number increased. Okay, but after industrialization, when industries came, lichens are a good indicator of pollution. Okay, so when industries came, pollution increased, lichens vanished. So when lichen vanished, all the predators could see the white-winged moth and predators could not see the dark-winged moth. So here what happened, students? The dark-winged moth increased in number and the white-winged moth decreased in number. Understood? This is what happened. And students, same like that only, we also have something called as anthropogenic activities. That means because of man-made, okay, like the use of herbicide and pesticides has increased so much that some organisms don't even die with that, right? Because the concentration has increased and they have become resistant to it. Okay, that is also a kind of natural selection. Now all of you notice cockroach in your home. Before, people used to put HIT, DDT or some kind of small solutions or acid, cockroach used to die. But now cockroach has become so resistant that even if you put HIT, cockroach will be like, oh, you're putting perfume, put little more for me. That is how cockroach acts. That is because these people have become resistant. These people have become resistant over generation after generation, and for them this dose is not enough to die. Understood? Storytelling, this is art. Art hai art. Okay. Understood? Asking understood only, my throat is paining. People won't tell by yourself, no. One job you have to put thumbs up, that also you won't. Okay. And like the video, that also you won't do. Nothing you will do, students. What you will do? Only you will tell ma'am which chapter to teach. Let me see. Is there any problem with the life? That you people are not responding. Am I stuck or something? No. Let's make it 100 likes, yeah. So shameful. Such a big channel cannot have 100 likes, sir. Okay, done. Now, frequency of AA, that is students, frequency of the dominant allele. Okay, this is important. Numericals can come. Frequency of dominant allele is P square. Frequency of recessive is Q square. Frequency of heterogeneous is 2PQ. Now, hence the formula is going to be P square plus 2PQ plus Q square. Also, if they give you a numerical and they have given Q value or they have given P value, you can use the formula P plus Q is equal to 1 and calculate it. Understood? P plus Q is equal to 1 and they can calculate it. Okay. So, change of frequency of allele in a population disturbs Hardy-Weinberg equilibrium. This change is due to evolution. Okay. Now, students, Hardy-Weinberg principle works in a proper ideal situation, that if there is a change in the natural selection, if there is genetic recombination, genetic drift, gene pool, if any such things happen, mutation happens, then Hardy-Weinberg principle will not be right, okay, will not stand right, because Hardy-Weinberg principle is an ideal situation principle. Clear to all of you? Now, very important graph. This is the last topic here, okay? Okay, come on. Yes. So, even I don't know the hours, dear. How will I know the hours? I don't know. Hope it finishes fast. Now, look here. Please try to understand this very simple. So, students, there is tall kids in the class, there is short kids in the class, and there is medium height kids. So, if the natural selection happens for medium kids, then that graph is called this one. The peak gets higher and narrower. This is called as a stabilizing graph. If nature selects the medium height people, then we will get a graph where the middle range is narrower. Okay, stabilizing graph. If the nature selects any one of the extremities, tall and short is an extremity, then you will get a directional graph. Okay, directional graph. But let's say nature selected both short and tall and did not select the medium, then you will get disruptive graph. If there is two, peaks like this, this is a disruptive graph. Understood? Same question they can ask you with, overweight and underweight and medium weight. So you should be having an idea that if the average weight is selected, you will get a stabilizing graph because the middle range is getting narrower. If they select any one of the extremities, it is directional. If they select both the extremities, it is disruptive. Clear to all of you? You asked me to go fast, I am going fast. Okay. Then students, this one you have to study. This I have put here for your purpose so that you can study. Now, how do you remember this? Just add the first all letters. Dra hen, like drag and no, dra hen. D for Dryopithecus, R for Ramapithecus, A for Australopithecus, A for Homo habilis, E for Erectus, N for Neanderthal man. So here I have just given you the values, so you can refer to this before going to the examination. See, brain capacity is important. Homo habilis 650 to 800 brain capacity, important. Same way Homo erectus 900 brain capacity, important. Match the following, they'll ask you. Then Neanderthal man 1400 cc. Okay, clear to all of you? Now here you see Homo sapiens is 1300 to 1600. So to confuse you, they can give you Homo sapiens and Neanderthal man. Now you will be like, huh? Homo sapiens come after Neanderthal man. So obviously their brain capacity should be more. But they have given you the lower limit. So this is the way they will trick you. So don't get tricked. Please write this, okay? Write Drahen in some side of the paper and see if you are in order. Because you should know that Homo habilis has 650 to 800, Homo erectus 900, then Neanderthal man, last only Homo sapiens. Clear? And who are the people who used to bury their bodies and also cover their bodies? It is the Neanderthal man. Okay? Fine. Java, all this is given here. This you have to read and understand. Done, done, done. Okay, I finished the chapter. Now, I'll call Basavaraj sir, 1 second. Because if I start with principles, I have to complete both. Okay, I have to complete both. So, principles I can't start. Principles and application at one shot I have to do. At one shot I have to do. Okay. Industrial melanism did not kill any species. Okay. Still all the species are there, just that the number decreased. Okay, the number decreased. Done, done, done. Clear? Any doubts? No, right? So, human health and diseases, you have to learn the immunity. Okay, I'll teach you the immunity part. And students, biotechnology principles also, I have the notes. Okay, sir will come in 2 minutes. So here, biotechnology, there's so many things that you have to remember very importantly, like PBR 322, selectable markers. So biotechnology principles will take us at least 1 hour. Okay, because it's a very like a lot of important concepts are there. Application will take very less time. Applications will take very less time. Okay, applications will take maybe like 20 minutes max to max, because I have to only teach you some terminologies. So it will take only 20 minutes. Clear? Brief account on evolution topic. See, that animal part and plant part is not that important because I did not see much questions from that. But evolution of man is very important. Okay, evolution of man is very important. So if you're not able to understand that brief account on evolution, you can just skip that topic and you can go to evolution of man directly. Clear? Yes. Yes, sir will come, just 2 minutes. Okay, just 2 minutes. I would have taught, but the problem is I can't stop it in the middle and go, no? It will be very awkward. So that's why I'm waiting. Okay, after this I'll come. After sir finishes ecology and principles of inheritance, I'll come with human health, biotechnology and applications. Clear? Have you shared it, sir? Short note. Yeah, PPT, yeah?
Yeah.
Want it in PPT only?
No, open it, no, click on it.
I'll get it down. Okay. One second. No. You put it on this one only. So when I come, I don't have.
What is there in this one?
No, only this human health is bad. Yeah. No, don't need. Roshni, no need. Horse evolution and all, don't waste your time. Last minute, no time. If you are finished it, good. 2 chapters of biotechnology after this. So what's the order?
Okay.
Microbes.
Thank you. So hello students, good evening students. I hope I'm audible and visible to each and every one of you. Good evening students. Quickly. See, microbes, nothing here students, don't have to worry. Microbes, they'll ask you mainly the examples which you all of you know. And the other thing which they can ask you for microbes is STP. If you want, I can teach you STP, it will not take more than 10 minutes. Okay students. Good afternoon students, good afternoon. Now, can we start the principles of inheritance? All of you. Can we start students? Can we start? Quickly in the chat students. Can we start principles of inheritance, all of you? Quick. Can I get a thumbs up in the chat? Can we start the principles of inheritance and variation now? Yes, all of you. Now I'll tell you exactly students what they'll ask you here, which type of questions will come and what you can expect the examiner to ask you. Okay, now can I ask all of you a very simple question now? The simple question is that, can anyone tell me who is known as the father of genetics? Quickly students. You will tell me, sir, father of genetics is called none other than the Gregor Mendel. Gregor Mendel is the father of genetics, right? So this entire chapter starts with Mendel and what did Mendel work on, and later on we go to non-Mendelian genetics, starting with Mendel right now. Okay. Now the first thing which you need to understand here is that what is inheritance. For example, students, how many of you heard in your life that people, your relatives will tell you that your nose looks like your mother, your eyes looks like your father? Does anyone tell you that, students? Have you ever heard that someone saying that your nose looks like your mother, your ears looks like your father, or your eyes look like your mother? They tell you that, right? They tell you that. That is because certain types of traits are transferred from your parent to the offspring. That is called as inheritance. It is transfer of characters are passed from your parent to the offspring. The transfer, that is called as inheritance. This inheritance is that transfer of traits. Now students, what about, are you exact copy of your mother and father, or are there any variations? For example, yes, my nose might look like my father, my eyes look like my mother, but my ears, my hair, my entire different facial structures, they do not look like any of them. It means that there are certain variations also occur when the characters are being transferred from the parent to the offspring. Those changes are called as variations. The chapter name is Inheritance and Variation. Principles of inheritance, all of you know. Transfer of characters. Variation means those changes are called as variations. Okay, now students. Mendel conducted a hybridization experiment on garden pea plant for 7 years and proposed the law of inheritance in the living organisms. So he worked on the pea plant, that is called as Pisum sativum, for 7 long years, and after working for such a long time, he gave us 3 laws. Can anyone tell me what are 3 laws, students, quickly? What are 3 laws? The first one is called as law of dominance, second is called as law of segregation, third is called as law of independent assortment. Now you need to know these laws for examination. I can ask you. Okay. Next question, students. Mendel selected 14 true breeding pea plants. What do you mean by true breeding? True breeding means that plants which has undergone self fertilization or self pollination, repeated self pollination, repeated self pollination. That is called as true breeding pea plants varieties, and 14 contrasting traits. So obviously, if you took 7 characters, he would have taken 14 traits. Now I'll teach you what is the difference between a character and a trait. Understand. Difference between a character and a trait. Understand, students. With the example I'll teach you. Let's take color of flower, right? Plant height. Now I'm talking about color of the flower, plant of height. Now this becomes a character. Why it becomes character? Because here I'm not asking any type of variations of it. This becomes a character. Now what if I tell you red color or white color, or I will tell you tall and dwarf, right? These becomes trait. They become trait now. They are the trait. Red, white, they are the trait here. Trait. Tall dwarf is going to be a trait. Plant height is going to be character. Similarly, flower position is going to be a character. It could be terminal or axial. Pod seed shape. Seed shape is a character. It can be round or wrinkled. That becomes a trait. Seed color, it is a character. It can be yellow, it can be green. That becomes a trait. So all of you know the difference between a trait and a character to some extent now, all of you. Now listen to me all of you carefully next. The true breeding lines is that, oh, it's gone written here. Continuous self-pollination is written. Okay. Now, the first hybrid generation is also called as first filial generation or F1 progeny. In a monohybrid cross, now students, monohybrid cross. Now what can they ask in examination is draw the entire monohybrid cross. For that, what do you need to do? You have to draw a graph, you have to draw entire table like this. Starting off with parents, dominant, tall, recessive, dwarf. Then capital T is going to be homozygous, small t is going to be also homozygous. Now what is heterozygous then? Heterozygous is going to be capital T, small t. This is heterozygous, and this we will not be using it right here. We'll be using only homozygous here. Then we get the gametes. Here. Capital T, small t. Then we have F1. F1 is going to be tall here. Can anyone tell me why do I say F1 is tall even though I have capital T and small t both are here? Then because of law of dominance. Because capital T here is dominant and small t is recessive in nature. So this right here is heterozygous dominant right here. Now what we do something called as selfing of F1. We take this particular right F1, cross it with itself, and we end up getting the F2. We can see a gametes capital T, small t, and we do a simple cross here. And we obtain the phenotypic ratio, genotypic ratio. What is the difference here? Phenotypic ratio, we can see here, students, tall, tall, tall. 3 are going to be tall, 1 is going to be dwarf. That is 3:1 ratio, right? 3:1 ratio. Then we have genotypic ratio, that is 1 homozygous tall, 2 heterozygous tall, 1 homozygous recessive. So 1:2:1 is the genotypic ratio. If you write this much, you will get all the marks. Nothing to worry in monohybrid cross. Okay. Now, according to Mendel, characters are controlled by discrete unit called as factors. So, see, now we call them genes. Now we call them genes. Back in the days, Mendel called them factors. And all of these factors control all the different types of characters in us. So, right now we know it as genes. Okay. For a character, plant height, capital T and small t are called as genotype, right? While means tall and dwarf. Now, one question arises, sir. What is alleles now? What is an allele? Allele, understand. For example, there is a gene here for tallness. It is capital T. Now, this particular gene will have an alternative form, like a twin brother or twin sister. Okay, so it becomes capital T. These are the alleles for it. So, one gene has two alleles here. Now, can I ask you a simple question? Students, is it possible that one gene can have more than one alleles? Students, quickly tell me. Yes, it is using test cross. Can anyone tell me in the chat, do you think one particular gene can have more than two alleles? Yes, it is possible. The concept is called as multiple allelism. We learn that in codominance. Coming up, don't worry, it's coming up. Okay. Now, again I told you this is homozygous, this is heterozygous. Okay. Now, students, what is Punnett square? See this. This right here is called as Punnett square. Right here is called as Punnett square. Right here. Okay. Now, what is test cross? Some of you might be thinking, sir, what is test cross? Why do we do test cross? Understand, it's very simple. Test cross is very simple. Understand what is test cross right now. Okay. Now, there is something called as, first of all, back cross. Right? There are two types of back cross. When we take F1 and we cross it with a dominant parent, that is called as an outcross. It is not given in your syllabus. When we take F1 and we cross it with recessive parents, that is called as a test cross. That is cross between dominant phenotype. For example, okay, I am taking a pea plant height. We have tall and dwarf. Okay, we have tall and dwarf. Which one should I take, students? For test cross, which should I take? Should I take a tall or should I take a dwarf? If I am doing a test cross with a plant height, what should I take? I should take a plant which is very tall here. So, a dominant phenotype we should take. Okay, dominant phenotype with recessive parents. So, we are taking a dominant phenotype and crossing it with recessive parents. Okay, why do we do it? To find the genotype of the F1. To find the genotype of the F1. We take the dominant phenotype. For example, see here, students. In this diagram which is given in NCERT, we have a dominant phenotype. See, for example, flower color. White is recessive, violet is dominant. So, we took a dominant phenotype here. Now, this dominant phenotype here, the genotype can be capital W, or it can be capital W, small w. Yes or no? It can be both. It can be both of them, right? See, this also can give you purple color flower. This can also give you purple color flower. Why? Because capital W is dominant and it can give you purple. Now, the whole point of your test cross is to find out which is correct here. The whole point of test cross is to find out, is this correct or is this correct? Which of the genotype is giving me this color? Which of phenotype is giving me this color? That is called as test cross. So, we are doing testing here. We are doing. In testing, what are we doing is, we are taking the F1, there is the F1 plant here, right? And we are crossing with recessive parents. Why? Because this F1 can be this or it can be this. Any of the two is possible. Okay? Any of the two is possible. It can be both. See, it can be this also, because it is purple, this also. Now, when we do a test cross, for example, here we took small w, here we have capital W, small w. So, when we do a test cross, we will notice that, wait a minute, when I am crossing small recessive with this one, I am getting 2 white. When I do a cross with double recessive here, I am getting all purple. It means that to find the genotype of F1, we need to look at the phenotype of F2. Based on this F2 and this F2, I can tell what is the genotype of here. If I am getting white color, it means that somewhere a recessive allele is present. Here. Okay. Now what about here? Capital W. It means that here, if I am taking all violet here, somewhere it is there saying that all dominant is present here. That is why I am only getting purple. It is purple. Okay. So with the help of this simple method, we can find which is going to be the genotype of your F2, genotype of your F1. So we're testing here. Okay. Madam, we'll do it in Kannada channel, don't worry. We'll do it. Tomorrow is the class. Okay, students. Understood all of you? This is called as a test cross now. Now, students, what is incomplete dominance? This is also a very famous question. They can ask you this question for 5 marks. They can ask you incomplete dominance, entire incomplete dominance they can ask you. Entire incomplete dominance they can ask you for 5 marks. Okay. Very simple, students. What is incomplete dominance? For example, let's take, I have 2 alleles here. Capital R, small r. I have 2 alleles here. Capital R, small r. I will do it, don't worry. Microbes, I'll do it if you want. Okay. Capital R, small r. Now, students, in this capital R, small r, can anyone tell me which is dominant here? Anyone who can tell me which is dominant here? Is capital R dominant or small r dominant? All of you will tell me, sir, capital R is said to be dominant here. But what exactly happens? The reality is not like that here. In the case of incomplete dominance, what happens is, right, this capital R is actually incompletely dominant. It's like, I don't want to be dominant. I just don't want to do work. Irresponsibilities. Completely irresponsibilities. Okay. So it is not going to be dominant over the small r. So when capital R is not dominant completely, what will happen? This small r also will start to express. Like, I will also talk. You know, students, you might have noticed that in a classroom, who makes the more noise? Backbencher or middle bencher? Students, tell me. Backbenchers make a lot of noise. Okay. Now imagine a classroom where backbenchers are very innocent. In that class, what happens? Even the first benchers will be like, I will also talk. They will also start talking that time. That is what is happening here. Okay. So capital R is incomplete dominant. So small r is also able to express itself. Here, students, small r. So in such case, what happens? One allele is incomplete dominant over the other allele. The F1 does not resemble both the parents. For example, students, let's say one parent is going to be red, one parent is going to be white, and the offspring here is going to be capital R, small r. This is going to be pink. It is not like your father also, not like your mother also. The child is very unique. Father is right. Father is a doctor, mother is an engineer. The son is a, let's say, I'm like, I don't want to be doctor. I don't want to be engineer. The son will be like, I will become a painter. Okay. So very unique it is. Example, it happens in dog flower, snapdragon, or atrium species. Anywhere it happens. It happens anywhere. Okay. Now this is a cross which you need to write. This is a cross which you need to 100% write. Pakka you have to write this. Same thing you can follow in examination. Parents, red flower, white flower. Gametes, capital R, small r. Then we get the gametes here. Gametes we get capital R, small r. Okay. Then F1, what do? We cross them. They get a pink color flower. Then we do selfing of F1, capital R, smaller, Punnett square. And once we do a Punnett square right here, we get What do we get? Red, pink, pink, white. So 1:2:1. Genotypic ratio 1:2:1. So can I tell here phenotypic ratio is equal to genotypic ratio here? PR is equal to GR here. So that is your incomplete dominance found in dogflower, snapdragon, or miraculous jalapa or anywhere else. Okay. So all of you write down this. Don't worry, you will get all of these notes in the Telegram. In Telegram, I will upload today only. Telegram is right here. I'll upload today only in Telegram. So download from Telegram today only. Okay. Incomplete dominance, done. Incomplete dominance, done. Okay. Now students understand here, when we did monohybrid cross, in monohybrid cross, we get two laws actually. We get law of dominance as well as law of segregation. Law of dominance and law of segregation both are obtained from your monohybrid cross. Okay students, so how many of you want me to explain law of segregation one more time in the chat quickly? How many of you want me to explain the law of segregation? Because law of dominance all of you know. Law of dominance all of you know very clearly, all of you can tell law of dominance very easily. Right? Very easily all of you can tell law of dominance. Do you want me to explain law of segregation? Tell me yes or no quickly in the chat because it doesn't take 5 minutes. Or why is it called as law of purity of gametes? Why is it called as law of purity of gametes, and why there is no exception here? There is no exception to this rule. Yes? Want? Understand students, understand. Okay, how many of you tried to make Oreo milkshake in the house? Or how many of you tried making tea in the house? Tea. How many of you tried making tea in the house? Quickly students, tell me. Inbreeding and true breeding, right? True breeding, repeated self-pollination. Same, inbreed depression also may cause of main reason, that is only. If you tried making tea in the house students, imagine you add water, you add tea powder, you add sugar, okay, you add milk, you boil it nicely, and you get the tea. Now you tell me students, is it possible after you make the tea, is it possible to remove all the components out of it? Like, tea is made already. From this tea, can you remove tea powder separately, sugar separately, water separately, milk separately? Is it possible to remove everything properly out of it? Not possible, no? Not possible. Now, when it is not possible, right, that is called as blending. It is all blended inside. Like juice, when you make juice, blending, everything has been mixed there. Blending means mixed. Okay. So before Mendel, everyone thought that alleles or genes, right, factors that time, they show blending. Like once it is blended, it is lost forever. You cannot get it back. That is what everyone used to think before. But what is the reality? The reality was Mendel came and told that in law of segregation, according to law of segregation, alleles do not show blending. What do I mean by that? I mean by that is this capital T small t is there, right? They are not blending completely. They are present as individual entities still. They do not show blending. Now, what is the proof there? What is the proof that they are not blending? Very simple. Now, you tell me students, if capital T and small t, they are blending very well, how do we get small t again back here? If in the F1 generation, we got tall plant here, and according to blending, dwarf is lost now. How did we again get dwarf here? It means that alleles do not show blending. Right? Alleles do not show blending. They segregate during gamete formation. For example, this capital T small t is here. This capital T small t here, students. Right? This capital T and small t is here. During gamete formation, they segregate into gametes. Alleles segregate into gametes. That is called as law of segregation. It is also called as law of purity of gametes. Why? Because this gamete is pure. It is pure for tall. This gamete is pure for dwarf. One gamete is pure for dwarf. That is called as law of purity of gametes or law of segregation. Okay, so they are segregating during gamete formation. They do not show blending. There is no blending here. That is law of segregation. Okay. According to law of segregation, the factors or alleles of pair segregate from each other. When they are in a pair, they segregate from each other during gamete formation. They segregate from each other during gamete formation. And each gamete receives only one factor of the character. They will receive only one. This gamete will receive only one of the factor here, receive one more. Okay. Income to dominance done. Also, income to dominance is done. Now, what is codominance? Understand what is codominance. In codominance, what happens is here in the example is human blood grouping. In human blood grouping, what happens is, students, one gene, I gene has 3 alleles here. They have 3 alleles. I gene has how many alleles? 3 alleles here we have. We have something called as IA, IB and small I. Now, what is the difference between all 3 of them? Very small difference is there. All the 3 alleles will produce different type of sugar. As a matter of fact, students, your I will not produce any sugar, but these 2 will produce sugar, right? Produce sugar which are different from each other. So 3 different alleles. Now, students, you tell me in the chat, just because I have 3 alleles here, can all the 3 alleles be present in one human? Is it possible that all 3 alleles can be present in one person? Is it possible? Out of these 3 alleles, any 2 can be present. 2 can be present. 2 can be present here. For example, let's take I have A blood group. My blood group is A. Okay. My blood group is A blood group. Okay. So my alleles are going to be IA, or it is going to be IA small I, because this is a dominant, so A will express. Now, what if my blood group is B? I will have either IB, both are dominant, homozygous, or this is my blood group. What if my blood group is AB? If my blood group is AB, it can be IB, nothing else. What if my blood group is going to be, let's say, is O positive? O. Then in that case, it's going to be, these are all different possibilities of A, B, AB and O group. That's all. You have nothing to remember here. Nothing to remember here. Yeah. Nothing to remember here. The grouping done. So, multipleism. What is multipleism, students? Multipleism is a concept where one gene can have more than one allele. One gene can have more than one allele. That's all. Nothing to worry there. Next, students. Now, we have 6 genotypes and 4 phenotypes. Okay. 1, 2, 3, 4 phenotypes. 1, 2, 3, 4, 5, 6. 6 genotypes. See, 6 genotypes and 4 phenotypes here. 4 phenotypes. In case of codominance, F1 generation resembles both parents, A blood grouping in the humans. So, in the case of F1, it will resemble both the parents. So, for example, if mother is a doctor, father is an engineer, the son is going to be engineer and a doctor both. Say biomedical engineer, son is going to be. Okay. So, both will be expressed in the offspring. In 19s, there are 3 scientists, Hugo de Veris, Kohn and von Schmuck, independently rediscovered the Mendel's result of inheritance of character. Very important scientist name. Rediscovered Mendel's work. So, now before we go further, students, now I will teach you something called as dihybrid cross. Ready, all of you? Not there, don't worry. Okay. Now I'll teach you something called as dihybrid cross. Dihybrid cross. Very easy it is. Dihybrid cross. Okay. First, what do you need to do? First, you need to do let's say parents, homozygous dominant, homozygous recessive, all recessive, smaller, smaller, small y, full recessive here. Then gametes, gametes done. Then we have F1, capital R small r, capital Y small y. R is dominant, so round. Y is dominant, so yellow. So F1 is going to be round and yellow, round and yellow. Okay. Now, what about selfing of F1? We take this particular one, cross with this one. Now, students, some of you might be like, sir, how do I write the gametes next time? Like for example, sir, as soon as I come here, I forget the gametes. How do I write the gametes? These gametes. Understand? Very simple. Very simple. Let's take capital R, small r, capital Y, small y. This is what we get in the F1. We get in the F1. Now, what you need to do here is this plus this. This. Okay. So, what do we get here, students? Capital R, capital Y, capital R, small r, capital Y, and small r, small y. So, we got the gametes. So, you need to write all of the four the gametes right here and do a simple cross. And when you do the simple cross, students, we generally obtain a phenotypic ratio called as 9:3:3:1 ratio. Phenotypic ratio. And let me tell you, students, they will never ask you genotypic ratio. Never. It is not there in your syllabus. Okay. Understood, all of you? This is going to be a ratio. 9:3:3:1 ratio we get. Phenotypic ratio. Okay. Now, let me ask all of you one more time, students. Do you want to learn how to get genotypic ratio? One small trick is there. You want to know? One small trick I'll teach you. How to get the genotypic ratio. You want to know? How to get the genotypic ratio, students. Want to know? Want to know, students? Okay, I'll teach you the trick, students. Listen to me. What exactly is phenotypic ratio, first of all? Right. Technically speaking, dihybrid cross is actually two monohybrid cross together. For example, in the case of monohybrid, students, can anyone tell me what is the phenotypic ratio in monohybrid? You will tell me, sir, phenotypic ratio in monohybrid is 3:1. So I told you dihybrid is basically two monohybrid. So take two monohybrids. 3 3s are 9. 3 1s are 3. 1 3s are 3. And 1 1s are 1. We got our dihybrid cross. Phenotypic ratio. Same you can do for genotypic also. 1:2:1 into 1:2:1. Do it. 1 into 1 is 1. 1 into 1 is 1. 1 into 2 is 2. 1 into 1 is 1. 2 into 1 is 2. 2 into 2 is 4. 2 into 1 is 2. 1 into 1 is 1. 1 into 2 is 2. And 1 into 1 is 1. That's all. Here is your students genotypic ratio. Here is your genotypic ratio of dihybrid. Dihybrid. How many of you understood? Nitin, don't worry, I will do it very soon in your Kannada channel also, don't worry. How many of you understood? How many of you understood the trick here? Very simple it is. If they ask you, you should be knowing. Okay. Once again, students, once again. Okay, students. Now, students, again, how many of you want to learn linkage and recombination? They'll ask you for 2 marks, right? Linkage and recombination, nothing more than that. By mistake, also, if they ask you something extra, you'll be able to write. Very, very simple concept, linkage and recombination. Let's start, okay? Linkage and recombination. Let's take, we have 2 people here, Rohit and Balaji. Those 2 are best friends. This Ravi and him, not so much, not so much. Now, what happens is, students, is that when same concept you need to apply in your genes also. For example, this is a chromosome right here. This is a chromosome right here. On this chromosome, when 2 genes are very close to each other, what will happen? When 2 genes are very close to each other, their linkage is very high, recombination is low. Recombination is low. When 2 genes are very far from each other, gene A and gene C, when 2 genes are very far from each other, what will happen? Linkage will be very low, recombination is going to be very high. So this basically tells you that, right? Basically tells you that linkage will always be higher when 2 genes are very close to each other. Like, you can hold hands, linkage is high. If they're very far from each other, linkage is not there, recombination is going to be there, right? Now, if you look at this particular diagram of your NCERT, right, you can notice here, right here, students, you can notice right here that, right, see, capital Y and capital W is present here, right. Now, can you see both of them, capital Y and capital W, both are very close to each other, very close to each other. So, you can see, students, linkage is very high, recombination is just 1 point, right, 1 point. Can't see, students, clearly. So, recombination is going to be very low. But at the same time, if you see this particular cross, W and M, the 2 genes are very far from each other, So what happens in such cases? Recombination is going to be higher, 37.2% higher here. That is a simple concept which you need to understand. Okay, simple concept which you need to understand. When 2 genes are very close to each other, right, linkage is always high. Very far from each other, linkage is low, recombination can be high here. Recombination can be high here. Okay. Now, Alfred Stewart went used the recombination frequency between the gene pair as a measure of distance between them and with the help of genetic mapping. So remember, students, if they ask you a question, who gave the genetic mapping? Your answer should be Alfred Stewart went. He gave the genetic mapping, also called a centimorgan, or the distance between the 2 genes on a chromosome. Distance between 2 genes on a chromosome was given by Alfred Stewart went. Okay. This cross is important, not really. They will not ask you the cross. You don't have to remember the cross here. Just remember what is linkage. Remember what is linkage. If they by chance they ask you, you should tell me the linkage is nothing but physical association of genes. Physical association of genes on a chromosome is called as linkage. That's all. Now, students, now we come to a topic called as polygenetic inheritance. Traits like human skin color. See, according to Mendel, we can only have tall plants or dwarf plants. According to Mendel, we can have green color or yellow color seed. According to Mendel, we can have round seed or wrinkled seed. It is always extreme with respect to Mendel. But in reality, that is not the case. In reality, what happens? Human height, for example, there is 6 feet, 6'1", 6'2", 6'3". There is 5 feet, 5'5", 5'7", 5'8", 5'9", 5'10". There is a gradation, right? There is a gradation in human height. Skin color, human skin color. There is nothing like light and dark. There is brown skin, light brown, dark brown, light, very fair albino color. There is a gradation in skin color also. All of this gradation is because of a concept called as polygenetic inheritance. Because of polygenetic inheritance. Now, what exactly happens in your polygenetic inheritance? Understand. In polygenetic inheritance, let's take, I have capital A, capital B, capital C, capital C. When such condition is there, a person will have very dark skin color. Now you see, capital A, small a, capital B, small b, capital C, small c. This is going to be brown skin color, brown skin. Small a, small b, capital B, small c, small c. This person will have very light brown color. Small a, small b, small c, small c. This person will have very white skin color, full cockroach, white cockroach, white cockroach. Okay, so you notice here, students, whenever there is, in the case of polygenetic inheritance, many genes control one character. What is happening here is many genes, gene A, gene B, gene C, multiple genes are controlling one character, that is skin color here. And it is effect here is additive effect. More number of dominant alleles, stronger is the effect. Stronger is the effect. Less, there is no dominant allele, less is the effect. It is an additive effect, and all students, this entire thing is also environmentally induced. You might have light brown skin, but if you go sit in sun, nicely when sun is coming, you do photosynthesis in sun, you sit in the sun the whole day, you will become dark. Tanning will happen. So this entire thing is environmentally induced also. Environmentally induced. Okay. That is polygenetic inheritance. Many genes control one trait or one character here. That is going to be skin color. Clear, students? Clear? Now, there is a concept called as pleiotropy. Pleiotropy is completely opposite here. I'll show you, students. This is done, students. Here, controlled by 3 or more genes. Right? One trait is controlled by many genes. Yeah. More dominant alleles, stronger is the effect. Next concept is called as pleiotropy. Pleiotropy is completely opposite. In the case of pleiotropy, one gene is controlling many effects. In pleiotropy, it's like a mother. Mother will do everything, right? Mother will, scold you. She'll feed you. She'll clean your room also. She'll wash the clothes. She'll make the food for you. Everything she'll do. Just like your mother, pleiotropy also explains. Always remember, MP, mother and pleiotropy, they're almost equal. Why? Because just like the mother, in pleiotropy also, one single gene can have multiple effects. One gene, multiple effects. That is called as pleiotropy. That is called as what? Pleiotropy. Example is phenylketonuria. Gene causing phenylketonuria. A gene that controls multiple phenotypic expression. Next is called as sex determination. Next concept is called as sex determination, students. Now, students, should I teach human here? Human is not needed, right? Human is very easy. All of you know humans, right? How it happens in humans? X, Y, what is there? Male will produce X or Y. If X is produced by male, girl. Y produced by male, you get boy child. You don't need that, right? All of you. Sex determination in humans is not required. Very easy it is. Now, what they can ask you here? There is something called as, I'll teach you honeybee. Honeybee is very important. I'll teach you honeybee. Don't worry. Now, there is something called as, students, male heterogamety and female heterogamety. Understand? Very simple. In the case of male heterogamety, it is the males which decides the sex of the offspring. Why? Because males have two types of gametes here. Take example of humans only. X, Y. X means girl, Y means boy will happen. So, male heterogamety done. Now, what is female heterogamety? Female heterogamety means the females here have two types of chromosomes. The females will decide the sex of the offspring. Example is in the case of your butterfly as well as your chicken. Chicken. Here, ZW and ZZ. See, in birds, female have ZW, so they have two different types of gametes, while the males have ZZ. So here, female heterogamety functions. That is in the case of chicken. Okay. Now, understand honeybee, students. Understand honeybee, very important. Honeybee, very important. Understand. What exactly happens in the case of honeybee? Okay. Understand, all of you. In the case of honeybee, right, number of set of chromosomes based on this, right, it can be a male or female. For example, males are haploid here, while the females are diploid. Males have 16 chromosomes, while females have 32 chromosomes. That number of chromosomes. Okay. All of you focus now here. In the case of honeybee, what happens? Male is already haploid. So male undergoes mitosis to produce the gamete haploid, while the female undergoes meiosis to produce the egg here. Why meiosis? Because reduction division has to happen here to change the ploidy. Whenever there is a fusion between the male and female gametes, it will always result in a female. Males are never produced as a result of fertilization. So once we get a female, it is going to be diploid, obviously, haploid, diploid, to n plus n, 2n. And this female can be queen or it can be a worker. She can be a worker. Any of the two possibilities is going to happen here. Now then you might ask me, sir, how are the males produced? Males are produced by a process called as parthenogenesis. That is, from the egg directly they will develop into male. No fertilization, nothing required. So from the egg directly they will develop into males here. Now you might notice here, students, there is no father here. Why? Because see this male has only one mother. It is directly developing from mother, no father. And this male, students, if this male produces the sperm, it will always result in a female only. If this male produces a sperm, it will always result in a female. So no son also. No father, no son. But there can be grandfather. Mean by that, see this male, right, it is developing from the egg. This egg is produced by a female. But this female, how did this female come into existence? This female came into existence because of a fertilization. And in fertilization we need a male here. So this male, though doesn't have a father, this female has a father. So male has a grandfather. See this male here, no father, but this female is developing from this father. So no father, but grandfather is there. See this male has no son, but this female can produce a son. Yes. So this male has a grandson. So here we can have no father, no son, but grandfather, grandson. All is the concept of honeybee sex determination. Female is always produced as a result of fertilization. Male is always produced as a result of parthenogenesis. Then students, then all of you. Then easy, no? How easy is it? Tell me. Easy, right? That's all is the sex determination in the case of honeybee. That's all it is. Very easy it is. Take a screenshot now only. Telegram I will upload after the class only. I will upload it. Yes, after the class itself I will upload this entire PDF. Okay? What is mutation? Mutation is nothing but sudden inheritable change. There can be point mutation. Can anyone give me the example of point mutation? Anyone who can give me the example of point mutation? Anyone? Point mutation? Very simple example is sickle cell anemia. Sickle cell anemia is example of point mutation. They can ask you the question. Okay. What are mutations? Mutations are the chemicals which produce the mutation. In pedigree, students, I want everyone to remember this chart 100%. Every single diagram you should know in this. You should know. They can ask you for 1 mark or 2 mark. They can ask you this entire thing. They can ask you. Okay. Now here I will not go very detail in the diseases because I have done it multiple times, students. If you want to know the diseases right in detail, please watch my lecture on principles of inheritance, where I have done, it could be line by line or it could be RNA series, right? Please watch my lecture on principles of inheritance. I have done in detail every single disease, but now I will just tell you the overview of it. In examination, one disease is pakka. I will discuss more in detail the 3 chromosomal disorders today. Okay, see this. Myotonia dystrophy is an example of autosomal dominant. This is not given in your NCRT lines. It is given in a NCRT pedigree is a pedigree chart. Legend says this. Legend says this. Sickle cell anemia, thalassemia, phenylketonuria are examples of autosomal recessive. Hemophilia and color blindness are sex-linked recessive disorders where heterozygous female may transmit the disorder to her sons. Okay. Now, students, do you want to do a question here? Do you want to do a numerical here? Color blindness numerical you want to do? Do you want to do a color blind numerical? Tell me yes or no. Color blind numerical you want to do? I'll give you a numerical. Okay. Numerical is very simple. Let's give a numerical. What percentage of offsprings are colorblind if normal male marries colorblind female? I'll answer. What percentage of offspring are colorblind if the normal male marries colorblind female? How do we do it? Very simple. Normal male, right? So he's going to be XY. Colorblind female, right? Colorblind female. Let's do a cross. XY. So answer is, what is the answer? 50% are going to be colorblind. 50% are going to be colorblind. I'll modify the question. As well. Marries a normal female whose father was colorblind. Tell the answer now. This normal male, right? Female, what do we have now? Father was colorblind. XCY. This X chromosome came here. So she becomes a carrier right now. Carrier. The answer is 25%. Okay. 25% of them are colorblind. Okay, I'll ask you one more question here. What is the, I ask you the question, what percentage of not tell the answer. What is the answer now? Here we can see, students, this is your 100% male, 50% is normal, 50% is colorblind. So this becomes 50%. 50% now. Understood all of you? Sir, we do not know information about females. Don't worry, no. If information is not given, assume it is positive. If it is not given, just assume it is positive. Okay. Here it will be 50% now. So 50% is normal, 50% is affected here. Okay. Understood all of you? Understood? Understood? Here 50% now, because entire thing is 100, entire thing is 100, that is for offsprings. Now the question has been changed to males now, males. So when you talk about only males, this you don't have to include at all, not include at all. So this is 100% now, this entire thing is 100%. 50% is normal, 50% is colorblind. 50 is the answer. Understood all of you? Understood? Katir, understood? How we got 25? Previous question was 25 because it says full offspring. It said full offspring. That's why if it is full offspring, what happens in students? If it is full offspring, what happens? This is normal, normal, carrier, and this is colorblind, right? Colorblind. Entire thing is 100, right? And this is going to be 25%, 25, 25, 25. 25% is affected. That's all. Okay. Female can be carrier, no? Who told she can't be carrier? Female can be carrier here. Okay. You have to see, every single time the information is not given, you have to assume it is positive itself. Okay. Take it a pinch with a salt. Same question, students. Same question you can also get for hemophilia also. Same type of question you can get for hemophilia. You should be able to answer very easily. Nothing difficult here. Very important, sickle cell anemia, students. Sickle cell anemia is very, very important. Sickle cell anemia is caused by substitution of glutamic acid by valine at 6th beta position. 6th position of the beta globin chain. Why is phenylketonuria? What is the main reason for phenylketonuria? The main reason for phenylketonuria is lack of enzyme. If enzyme is not present, right? Enzyme is not present, what happens? If enzyme is not present, individuals cannot convert amino acid in the alanine to tyrosine. Phenyl hydrolase enzyme is not there, tyrosine will increase in the blood. Sorry. Phenyl alanine will increase in blood, it will get converted to pyruvic acid, and you will have lot of acid in the brain, in the muscles, in the entire body. Very disease. Thalassemia, we have two types, HbA1, HbA2. Chromosome 16, the parent. What is aneuploidy? What is polyploidy? Right. In aneuploidy, what happens? Gain or loss of chromosome, increase in number can happen above or below. Polyploidy is increase in set only. Set happens. Or euploidy. Like for example, can become from 2n, it can become 4n directly. Last topic, 3 types of syndromes are here. Now there they can ask you. You don't take both the possibilities, you take only the positive possibility. Like that's how the question works, right? Question usually works with the positive possibility. When it is not given, you always check it as positive and you do solve the question. Okay. Now this is the last topic from my side today. This chapter. After this, I'll come and I'll finish ecology, students. Ecology is very easy. Entire ecology I can finish in 30 minutes or 40 minutes. Entire ecology will happen. I'll tell you what you need to study, what you don't have to study in ecology directly. Okay. In Down syndrome, what happens? In the case of Down syndrome, there is one extra chromosome or one less chromosome. In Down syndrome, there is one extra chromosome. It is called a trisomy of 21. Trisomy of 21. Now here, symptoms are not given. You have to remember symptoms. For Down syndrome, what is symptom? Broad, round head, furrowed tongue, right? Retardedness of mentalness, mental retardedness, psychomotor development is not there. Broad palm with characteristic creases. So all of that you have to remember. Down syndrome. Klinefelter syndrome. What happens? In the case of Klinefelter syndrome, there is one extra X chromosome is there. It becomes XXY. Because of this extra X, what happens? There can be development of breast in the male called as gynecomastia. gynecomastia. Okay. Now, what is the Turner syndrome? In case of Turner syndrome, one X chromosome is missing there. One X chromosome is missing from here. Okay, so it becomes XO. And because of that condition, what happens? The females are sterile and very short stature and rudimentary ovaries. You have to remember these symptoms at any cost. Okay, students. Now I'll call Gopika ma'am for human health. Or do you want or you don't want human health? Students, do you want to know human health?
Hello. Are you all there? Tired? So strong, my kids are. My God. Rihanna, who I can see from morning class. Mohan Raj. Wow. Power packed kids. They're there from morning. They're like, ma'am. Okay, students, I want to tell you people, all of you, one thing. Just let me know if I'm stuck or not. If there is a lag, there's no lag, just let me know here. Second, let me just upload the PPT. Hello, welcome back. You guys missed me? Okay, 1 second, just uploading the PPT. So every, 5 minutes we are going, making PPT and coming back. So this is the workload that we have. Okay. Yes. No lag. Yes. Okay. No lag. Students, I wanted to tell you all one thing. So I'll be posting a video very soon on the channel. It is a long video, but the thing is you can selectively watch it because it's going to be uploaded. You can watch it at your own speed. In that video, I will be doing principles, then application, as well as human health and diseases, AIDS and cancer in detail. Okay. So for those students who wants to watch biotechnology can watch it from that video, because now in this video and that video I have taught the same thing. So why to just repeat it? Okay. Are you all okay with that? So the video will be uploaded very soon. Okay. So that you people also get some break, even we get some break, because we have to do questions also after this. So that is why we need some more time to collect all the questions. I have collected around I think 160, 70 2 marks questions, 3 marks questions. So I have collected everything. So we want to show that to you people. So is that okay with all of you? Biotechnology principles and applications will be in the next video. That means it is going to be uploaded in an hour or so. So you can watch it from there. There I have detailedly explained, like drawing the diagrams and everything, and you can watch it at 2x. Okay, because here you cannot watch it at 2x. You have to bear with me in this speed. Okay. Yes. Yes. Thank you. That's so sweet. It will be easy only, yaar. Paper won't be that difficult. Just the paper tricky part will be just the MCQs. Okay, MCQs will be the little tricky part. And let me tell you, with CBSE, what is the thing? They change the way of the articulation of the questions. Okay, they change the articulation of the questions so that the students get a little confused. Okay, if you see your previous year questions, you will understand. Like, instead of directly asking Lamarckism or directly asking Darwin, they will give you one giraffe image and they'll show you, you know, the giraffe neck length increasing, and and students, yes, evolution also is detailedly in that lecture. Okay, evolution also is there. Whatever, like, I'm not able to do here because, you know, we are standing from morning, we are a little tired. So that's why I thought, okay, I'll just upload the biotechnology videos. There also I've done detailed lecture, beautiful images are there, better, you know, presentation is there. Okay. Yes, and also you can watch it at 2x at any time. Even at midnight you want to watch it, you watch it. Because live what happens, that is not possible, no? Okay. So after this, so what's the flow from here? After this, sir will do ecology in half an hour or so, and then we will end this session, and then we will meet you in most important questions. We will neither make you solve few questions or we will give you the PDF of the questions. Okay. Done, done, done. Because after this we have one more KCT class on Vedantu Karnataka channel. So that's why if I take biotechnology now, again I need at least 2 hours. So then it will become 6:30. Then you will not have time to prepare for anything. Okay. Yes. Okay. So that is a very long video, but you only selectively watch what you want, because it's starting with biotechnology principles, then it is human health, and then it is evolution. Okay. Yes, clear? Students, yes, let me check that also. If applications is not there, I'll finish off applications here. Okay, otherwise you people will miss out on that, no? I mean, I have videos for everything. It's not like it's not there. But just, yeah, I'll check that and I'll get back to you people, okay? In case applications, I think it's not there. So we will do applications also now. Chalo, let's start. Okay? Ready? I think both is this. 1 second, sorry, so sorry. Okay. Oh, there's no net. That's not. Okay, we will start now. Okay, are you all confident now? How is it going? Are you all confident now? Yes or no? Yes, ma'am. No, ma'am. That. Okay. Yes, application also is there. Okay, application also is there. Okay, don't worry. Both are there. Okay, chalo. Let's start. Human health and diseases. Now here, from question point of view, I have seen a lot of questions on pneumonia. So let's just also revise typhoid. Okay. Typhoid is caused by Salmonella typhi. Typhi. Easy to remember, no? Salmonella typhi. Okay, but still stressed. Don't worry. Don't be stressed. Be calm. Exams are going to happen really well. Okay, don't be stressed, because if you are stressed, you will forget everything and then unnecessary problems will come. So just relax. Be like, okay, both I can manage it. Okay. Yes. So typhoid pathogen is Salmonella typhi. Mode of transmission, it enters the small intestine, okay, through food and water. So students, from contaminated food and water, typhoid will be caused. All of you know this, I believe. Okay, typhoid is caused from food and water and migrates to other organs via the blood. So symptoms, very important symptoms are, the fever is going to pertain from 39 to 40 degrees Celsius. Okay, it is going to be sustained fever. That means in malaria, you will read the word across, recurring fever. Recurring means what? Like, you know, like your guest, every time they don't come to your home. They come once in a while and go back, right? Recurring means like that, 3 to 4 days, once in a while the fever comes. Sustained means throughout the fever will be there. Okay, 39 degree to 40 degree. Headache will be there, weakness will be there, stomach pain will be there. Okay, because constipation, everything is with the intestine, right? So you will have constipation, you will have abdominal pain. Okay, sometimes even diarrhea, depends. To people, it will be little different. Now students, in very, very severe cases, there will be intestinal perforation. Okay, very severe cases, there will be intestinal perforation. Vidal test is used for confirmation of the disease. Very important. Okay. Now, I'll tell you about one lady whom you might know, Mary Mallon, or Typhoid Mary. She had typhoid and she was a cook in many households, okay, and every time she was falling sick, she used to never, like, you know, take rest. Like our parents itself, like we tell our mother, no, just mama sit here, don't do any work. She'll be like, no, I am totally fine. Okay, I can be the Usain Bolt, I can run all, you know, I can go for Olympics also. But like that only Mary Mallon used to ignore all the symptoms that was there, and she used to continue, you know, working in different places, and every single place she went, she contaminated the food, and people died because of that. Okay, people died because of that. So we call her Typhoid Mary, and she was a carrier, human carrier of disease. Human carrier of disease. Okay, yes, okay. No, typhimurium, same thing. Short form we write Salmonella typhimurium typhi. Okay, pneumonia is caused by Streptococcus pneumonia and Haemophilus influenzae. Streptococcus pneumonia, just now you people studied in Griffith's experiment, Streptococcus pneumonia and Haemophilus influenzae. Okay, it affects the lung alveoli, and the alveoli gets filled. This is important. The alveoli gets filled with fluid. Because of it, the person will have breathing issue, like the person will not be able to breathe. Okay, so here students, inhaling the droplets or the aerosols. Now let's say example, X person is standing here, he sneezes, he has pneumonia, I am standing next to him, the aerosols, I take in the aerosols, I get pneumonia. Okay, because in that aerosols what will be there? Your Haemophilus influenzae will be there, or Streptococcus pneumonia will be there. Okay, yes. Now students, not only that, sharing the pen, the utensils, now the water bottle I am drinking. Okay, the water bottle I am drinking also, we will get disease. Okay, like that only I will drink some water also. Yes. Okay. Mode of transmission, inhaling the droplets, aerosols released by an, Roshni is here. Okay. Infected person, sharing glasses and utensils. Symptoms, students, respiratory problem. Most importantly, fever will be there. But what is the most important? In severe cases, the nails. Okay, the nails and lips will turn bluish in color. That is very important to remember. In severe cases, lips and nails will turn greyish to bluish in color. Okay, done, done, done. Okay, done, done, done. Now, common cold. Who doesn't have common cold? Every alternative days, once at least common cold should come. Otherwise, we'll be like, oh my god, my immunity is better or what? Like, suddenly, why am I not getting cold? So, common cold is caused by rhinovirus. Okay, it's a virus, rhinovirus. That is why we never get rid of cold, because virus every time comes in different variants. That's why body will not be able to hold the memory of it. Okay, mode of transmission, students, all of you know how you get cold, by taking in the droplets, touching the utensils, or sharing the, you know, things, we will get immediately cold. Okay, anything like computer, keyboard, whatever, phone, everything will get. Now students, symptoms are nasal congestion, you know, you will not be able to breathe properly. At the same time, discharge will be there, runny nose will be there, sore throat will be there, throat infection can also come, and common cold will last. That is, the incubation is for 3 to 7 days. Incubation is for 3 to 7 days. Done, done, done. Yes or no? 3 to 7 days. Okay. Good evening. Yes, protozoan diseases, students. Malaria, very important. Malaria and pneumonia is asked a lot for CBSE examination. Pathogen is Plasmodium, Plasmodium vivax. In that, question can come, which is the most malignant one. That means most fatal one is, Malaria falciparum. Okay, falciparum is the most fatal. That means death can happen. Okay, death can happen. Now, most serious, that is malignant malaria. See, malignant one is falciparum. Mode of transmission, students, by female Anopheles mosquito. Okay, Anopheles mosquito. Clear? From good morning to good evening, you people are seeing me, no? My students saw me first in the morning. Now, in the last day of the end of the day also, they'll see my face only. Okay, morning they saw me at 10, I think at 10 o'clock they saw me. Now they'll see me in night 9 o'clock also. So yes, but yeah, mode of transmission, like if you have students like this, you always feel energetic. But if you have students who are like complaining, ma'am, go fast, ma'am, when will you finish, ma'am, what is the duration, ma'am, will this come for CBSE, ma'am, then we'll feel like, okay, we should just leave the students and go. Okay, yes. So, students, symptoms are going to be, yes, very important. I told you that the symptom for malaria is going to be recurring fever. Okay. Recurring fever. Recurring means what? Every 3 to 4 days, the fever will come back. And this is because, students, at the cycle, there is a release of a toxin which is given in your NCERT called as hemozoin. This hemozoin gets released and immediately you will have fever and chills. Okay, fever and chills. Clear? Yes, fever and chills. So that is why we get recurring fever. Okay, recurring fever. Yes. Yes, Basavaraj sir too. So this is understood, no? So every 3 to 4 days, fever will be there, chills will be there. Okay, fever and chills will be there. Now students, life cycle. Okay, life cycle of your mosquito and human. So understand that your malaria needs a digenetic host. Digenetic host. That means they need two hosts. Okay, man and mosquito. Man can be like an intermediate or a secondary host. Primary host is always going to be female Anopheles mosquito. Okay, female Anopheles mosquito. Now look here, look at the life cycle. What is going to happen, students? Sporozoites. Okay, sporozoites. Yes, once a while. Okay, so I thought Roshni is telling once a while. Okay, something. Okay. So sorry. Okay, students. So mature infective stage. See, sporozoites. Okay, sporozoites escape from the intestine. That doesn't matter. You just write the sporozoites. Okay, like let's say mosquito bit a healthy person. The sporozoites got into the blood. Okay, now from the blood, where will it go? It will directly go into the liver. From the liver, where will it go? It will go into the blood, that is into the RBC. Now, in the liver, what are they going to do? Ma'am, in the liver, these people are going to burst, that is, they are going to do asexual reproduction. Now, you can ask me, ma'am, why do they need two hosts? Because one will be asexual, okay, one is for their asexual phase, okay, very luxurious people, one is for their sexual phase, okay, one is where they can fuse with the gametes, one is the asexual phase. Clear? Yes. Yeah, the common cold occurs continuously. It's because of variants. Okay, if different variants will come to me. Okay, now parasites reach the liver. In the liver, what do? They multiply, burst open. Okay, and then they will go to the RBC. Okay, they will go to the RBC, and in the RBC also they will do the same thing, and they will end up releasing hemozoin. Spelling is wrong. Hemozoin. Okay, they will end up releasing hemozoin that causes the cycles of fever. Remember this carefully. Okay. Thank you. That's so sweet of you, Christina. Okay. Yes, asexual reproduction is by when the RBCs burst open and hemozoin will be released. So, very simple thing happened. Sporozoite was there in the mosquito. Okay, mosquito bit a healthy person. The sporozoites went directly into the liver. From the liver, it burst open. Asexual reproduction, continuous multiplication. It burst open the liver cells or the hepatocytes, and it went into the RBC. In the RBC also, there is erythrocyte cells are there. No? So, in the RBC also, they burst open the RBC, and from there they directly went to the release of a toxin called as hemozoin, which causes cycles of fever. Now, from here what happens? We cannot do sexual reproduction, but we can help in the formation of gametes. So, male and female gamete is in our blood. Another mosquito or the same mosquito can come and take the blood, and in that blood meal both the gametes will be there, which will fuse and form again zygote to sporozoites, and this cycle will continue. Can you understand something? Huh? So, gametocytes will be there, that will be taken up by another mosquito with the help of a blood. Okay, sucks the blood with the gametophytes, and then fertilization and development will happen in the mosquito's gut. Yes, MCQ question. Where does fertilization happen? In the mosquito's gut. MCQ. Where is the sporozoite stored? In the salivary gland. Storage. By end of the day, my spellings will go. Okay, storage of sporozoites. Storage of sporozoites will be in the salivary gland. The fertilization will happen in the mosquito. Okay. Khatar, good question. Yeah, we should ask mosquito about this. I have thought about it, but yeah, that is because most of our, like, in the sense whatever happens, no, it directly affects our liver, right? So that is the reason. Okay. Yes. Chalo. Let's do Ascaris. Okay. Ascaris, students. Pathogen is Ascaris. Mode of transmission Ascari. So, Ascariasis is the disease caused by Ascaris, which is an Ascalminthus, roundworm. Okay, now this is also from fecal matter. That means contamination has happened. Okay, the feces has somehow reached the vegetables or the soil. Now, how does this happen, ma'am? Let's take an example. Raju is there. So sorry if anyone. Okay. Raju is there. Okay, he has Ascaris. Okay, he has Ascariasis. Now, he has put the fecal matter on the soil. In this fecal matter, there are a lot of eggs, and later, after some time, let's say, Mintu buys it. Okay, Ayoli. Mintu buys this land. Okay, Mintu buys this land, telling that I want to do farming. Now, what will happen? This fecal matter is there. Obviously, it has got absorbed into the soil. The eggs of the parasite also is there. Now, let's say I grow cabbage. What is this? Okay, I grow cabbage. Okay, I grow cabbage, and it has come to the, to the market. Now, let's take example, someone consumes it. Okay, now someone consumes it, automatically that person will have Ascaris. This is the cycle. Okay, this is the cycle. Clear? Yeah, I sacrificed myself to teach you people by telling that I bought the property. Okay? Okay, no problem. Now students, the symptoms: internal bleeding, muscular pain, fever, anemia and blockage of the intestinal pathway. So this is little severe, okay? Internal bleeding will be there. At the same time, muscular pain will be there, fever will be there. Okay, next one is elephantiasis or filariasis. This is caused by nematodes, right? Here students, pathogen is filarial worm, that is Wuchereria bancrofti or Wuchereria malai, okay? Okay, Wuchereria malai. Mode of transmission, mosquito. Students, here also Culex mosquito is the person who causes it. Now what happens? It is the swelling of your lymph nodes and it is a chronic disorder. Like, okay, so filarial worm lives in the lymphatic vessels. Okay, it lives in the lymphatic vessels. It causes a chronic inflammation in of the organs in which they live for many years. Limbs and genital organs may be deformed. Okay, have you seen the image? You will see that the leg has swollen. It has reached till the genital area. So it is important for you to remember that who causes it. It is Wuchereria bancrofti or Wuchereria malai. It is a filarial worm. So we also call it filariasis. What happens? This worm will stay in the lymphatic vessels and they will do the swelling of the lymph nodes, because of which it becomes a chronic inflammation spreading till the genital area. Done? Okay, very easy chapter this. Okay, continue. Now, fungal diseases. Ringworm pathogens are Microsporum, Trichodermophyton and Epidermophyton. Okay, Trichodermophyton and Epidermophyton. They are seen in the groin. Okay, here these regions. Okay, where there is no light. Okay, like in the sense what I am trying to say is it's always covered. Okay, here. Okay, wherever there is sweat, like students, water can retain. Okay, groin area between the toes. Okay, between the toes. Now, mode of transmission, students, it can happen from a person who already has a fungal disease. Now, you go to gym. And you will see everyone is sweating, everyone are, sleeping on the same equipment and they're not cleaning, they're getting up and going. Now, what will happen? There is chances that we get the fungal infection. And how does it look like? It looks like a white lesion. Okay? Don't write white lichen, white lesion. Okay? It will look like this. Dot. Okay? Like this. It will look like this. Understood? Yes, ringworm allergy we tell. Okay. So, dry and scaly lesions on the nails, skin, nail, scalp, and there will be intense itching. Okay, intense itching. But here you should remember pathogens are Microsporum, Trichodermophyton, and Epidermophyton. Okay, Epidermophyton. Now, very important part, students: innate and acquired immunity. Ready, all of you? Done. Correct, Mohan Raj. I've seen a person, I've met a person who has elephantiasis. Okay, he was, he used to beg near one of the church in Bangalore. Okay, so I've seen him there. That time I used to not understand because I was too young that time. Okay, now we don't go there and all, no. So that time I used to not understand. I used to think that okay, God just made him leg very big. Okay, because I was very young, like I was in I think 2nd standard or 3rd standard, but I still remember how his leg used to look. So when I saw this image in my first PU, I was like, oh my God, I've seen this. Okay, yes. So students, immunity. Immunity means what? The ability of the body to fight a foreign particle. Now if a foreign particle is coming, body has to fight it. Yes ma'am, body has to fight it, otherwise there is an option or what? Okay, so body has to fight it. So that is called as immunity. Now 2 types of immunities are there. One that you are born with, one that you acquire during your lifetime. The one that you are born with is called as innate immunity, and the one that you will acquire later in your lifetime is called as acquired immunity. Yes or no? No, they will not ask like that. They will ask you to explain pneumonia, typhoid. They'll give you the name of the disease. You just have to write about the disease, or they'll ask you they'll give you the symptoms, and you'll have to find out what kind of disease it is. Most of the time, they'll give you pneumonia. Why? Because lips and nail stain gray to bluish. They'll give you, or they will give you Vidal test is used for which among it, and explain like that. They'll give you. Okay. Yes. Now students, what will happen? Yes, one that you are born with is, or one that you have from birth is called as innate immunity, and innate immunity is non-specific. That means it does not care if it is bacteria, virus, fungi, protozoan, helminths, nothing. It will be like, you come, I'll fight with you. It's like that. But then when you come to acquired immunity, they know that okay, I have to hit a virus like this. Okay, like how your mom knows by now, no, how you should hit you, how to do emotional damage, by now she knows, okay. So she'll ask you, did you study? That time emotional damage will automatically happen, or how much did you score, she'll ask you. Dad also knows one technique, no, what did I, where is the 100 rupees that I gave you last week? So like that, now they are very specific to how to attack you, right? So same thing, acquired immunity is more specific, that okay, virus have to affect like this, fungi have to affect like this, protozoans have to hit like this. But this fellow is not like that, man, just come, okay, come, like that. Okay, there students, it includes 4 types of barriers. This is non-specific. There are 4 types of barriers: physical barriers, physiological barriers, cellular barriers, and cytokine barriers. Okay, yes, physical barriers means prevents the entry of microbes, especially your skin and mucus coating. Students, mucus coating is where and all present in your respiratory system, digestive system, urinogenital system. Because from the nose, the bacteria can enter, mouth, it can enter through your what is that, urinogenital systems also, your microbes or any organism from outside, foreign particle can enter because there is an opening. So in all those opening we have mucus coating. Okay, all this opening we have mucus coating. So prevents the entry of microbes, skin is there, and mucus coated in the respiratory system, urinogenital system, and digestive system. Just remember that. Okay, yes, clear, yes. Now psychological barriers, they prevent the microbial growth like your gas, HCl, saliva and tears. Why you should cry once in a while is because dirt should go. Okay, we should kill the physiological barriers should work. Okay, so saliva in the mouth, HCl in the stomach, these are all as a preventive method so that we can prevent. Okay, next one is cellular barriers. There are some cells in our body who are going to do the job of phagocytosis. That is, if someone comes from outside, immediately they will go and engulf them. Okay, that is phagocytosis like WBCs or your PMNL neutrophils. Okay, polymorphonuclear leukocytes or neutrophils, monocytes and natural killer cells. Natural killer, okay, neutrophils, PMNL and WBC. Okay, WBC. These are the cells who will do the job like, oh, I saw you, I'm coming. Okay. Okay, yes. See, before I even tell, water is already coming. Emotional damage is already over. Cytokine barriers, students. Cytokine barriers is very interesting, but some students don't understand. Let's take an example. Okay, these are the 4 cells. Okay, now students, what will happen? Let's say in Suvra's home, if the current goes, what will Suvra do? She'll peep to Christina's home and check that, oh, if Christina doesn't have current, she'll be like, lucky, that girl's house also doesn't have current. But cells are not like that. Like how we humans are selfish, no, cells are not like that. Now let's take an example. A is getting affected by a virus. A is getting affected by a virus, okay, right? It is a virus infected cell. Now this virus infected cell already knows that my life is in danger. I should definitely save the other cells. Okay, I should definitely save the other cell. Now this A cell is a virus affected cell. It will release something called as interferons. It will release something called as interferons, which will help in signaling the B, C, D cells, telling that, you know what, A is affected by virus, you please save yourself. Immediately the B, C and D cell will become happy and they will form antiviral protein. They will form antiviral protein. This way what will happen? They will protect from virus. But will we do that? No, of course not, ma'am. We are humans at the end. Okay, but the cells inside us have this kind of mentality. So it protects a non-infected cell from further viral infection. Clear or not? Yes, clear. Interferons. Interferons will do the signaling of these 3 cells and these 3 cells will release the AVP, that is your antiviral protein. Now students, acquired immunity. So for those students who joined late, let me tell you that biotechnology and applications, I have uploaded another video. It is going to be uploaded in few hours. So that video you can watch it at 2x at your own speed. It's a very long video, but you only watch the portion that is required for you because it has biotechnology, applications and principles, human health and diseases and evolution. Here human health and diseases, I'll just teach you the outline. In depth, whatever is needed is in that video. Clear? Yes? Okay. That is only important concepts. It's not the entire chapter because the entire chapter though it will become some 20 hours marathon. The entire chapter is not the only important concepts from each chapter. Okay. Now, it is a pathogen specific immunity developed during the lifetime. So, acquired immunity means students, it is pathogen specific. I told you, they do not, they are very like, okay, I know, like pathogen specific immunity developed during the lifetime. It is characterized by memory. Okay, it is characterized by memory. Clear? Okay. Okay. So, memory, during the first encounter, a pathogen body produces a primary response. Students, when you first meet a person, you will be very shy and you'll be like, hi, how are you doing? Okay, like that. Okay. So, it's like, you know, when you call to a restaurant and you are trying to be very like, you know, very like classic person, and you're calling and saying that you want to order a sandwich. But what will you end up saying? Hello, I am sandwich. Can you order a Gopika? Like that. You know, that's like, you know, like, so you just change the name, right? So it's basically, hi, I'm Gopika, I want to order a sandwich, but you end up telling the ulta of it because you're scared, you're tensed. So even in body also, the primary response is not going to be that stronger, but the second time the pathogen comes, second time you go to that restaurant, you'll be very confident, you know what is this restaurant and what is going to happen, okay? So second time what is going to happen, students? Second time you will be confident. Same way here also, secondary response is going to be double stronger than the primary response. First time if body have sent 1000 soldiers, second time body will send 10,000 soldiers to kill that fellow. Okay. So, second encounter of the same pathogen causes a secondary response with higher intensity. Understood? Yes or no? Yes or no? Primary and secondary immune response are carried out by B cells and T cells. Okay, B cells and T cells are very important, and these B cells and T cells are related to your one more important topic. What is that? Forgot. I wanted to say this. Yeah, humoral. So sorry. Okay, obviously from 10 o'clock this much only brain will work. Humoral immunity and cell mediated immunity. Cell mediated immunity. Same thing, one more thing you have to remember is your primary lymphoid organ. Okay. Primary lymphoid organ. Okay, primary lymphoid organ is bone marrow. Okay, primary lymphoid organ is a bone marrow and thymus, right? Students, in this one more thing that you need to remember, ma'am, how many things are you making me remember? Bone marrow and thymus is very important. Why you know? Why you know? Yes, cell mediated graft rejection, humoral immunity is the B cells. Okay. Now, bone marrow and thymus is important because these people are going to help with the B cells and T cells maturation and differentiation. Even though T cells are going to do that in the thymus, bone marrow only will give the origin for both of it. Okay. Now, B cells produces antibodies and these proteins to fight the pathogen. So, remember that B cells have the bullet is the antibodies. T cells do not have the bullet, but they'll help. They'll tell, you know what, whatever help you want, I'll give you. Clear? Now, T lymphocytes, okay, T lymphocytes help B cells to produce the antibodies. Understood what I told? Now, here students, what is the most important thing? This is the heavy chain. The full Y is there, no? That is the heavy chain. This is the light chain. They are kept together with the disulfide bond, okay? 16 disulfide bonds are there. Heavy chain is there, light chain is there, and in between that, like a mouth, they have an antigen binding site where they will catch hold of the foreign particle. So, 2 light chains and 2 heavy chains. Types of antibodies is gamed. Okay, types of antibodies is gamed. Okay, game is there, no? So, same way you remember gamed. Okay, gamed. Thank you, Jai Ganesh. Gamed, remember, gamed means IgG, IgA, IgM, IgE, IgD. That IgA will be present in the colostrum, IgG will be the one who can cross the placenta. Okay, a NEET teacher can never stop teaching extra. Okay, yes, acquired immunity is of 2 types, active and passive immunity. Again, students, active means, see, let's take an example, Jai Ganesh and Roshni. Okay, Jai Ganesh and Roshni both are going for a party. Jai Ganesh went to shop, bought a ready-made dress. No, let's give it the character to Roshni, the girl. Okay, Roshni and Jai Ganesh. Roshni went to the shop, she got the material and then she stitched it. Okay, so she did not go for a ready-made one, she made a self-made one. Okay, she made a self one. Jai Ganesh, instead he went to Zudio, he bought a pant and shirt and he's like, okay, I'm sorted. So he did a ready-made one. Now you tell me who is active and who is passive. Obviously, ma'am, Roshni is the one who is active because she took the time to make the own dress. But on the other side, Jai Ganesh went for ready-made, so he is passive. Same thing happens with active and passive immunity. Active immunity is when the body itself produces the antibodies because of a subclinical infection. That means I got a fever, okay, I got some kind of infection, my body recognized it and my body is recovering from it, like what I had here, like an accident. So same thing. That is my body itself made the antibodies and cleared the foreign particle. On the other hand, you have passive immunity where we are trying to take ready-made, okay, ready-made, okay. Now, active immunity, students, it is in two types when active immunity happen, when we have a clinical infection, or second time is a vaccination. Ma'am, how vaccination? In vaccination, we give weakened antigen. That means a little like, unconscious antigen, okay, like an unconscious antigen we give. So artificial active immunity, it is developed by the injection of a microbe deliberately during immunization. That means we tell our body that, you know what, we have only injected the foreign particle, you please make, things against it. Natural is when you have an infection, okay, when naturally we get such kind of infection. Passive means, students, we have taken ready-made antibodies, we don't have time to make it, so we are taking ready-made. First one is when the baby gets the colostrum from the mother, IgA will already be present, but again, under passive also there is natural and artificial, here also natural and artificial. Why under passive colostrum is natural? Because we are not injecting anything into the mother's body, the mother is automatically making the colostrum, even though the baby is getting it as ready-made, okay. Artificial passive means, students, anti-venom, okay, anti-venom, because anti-tetanus, anti-venom. Why? Because our body does not have the time, like if we give the time for the snake poison to like get into our body and then our body make antibodies, by then we will be on the top. So that's why what we will do is we will inject it into the horse, and from the horse we will take the anti-venom and we will inject it into us. So this is ready-made, okay, done, 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 done. Okay, clear? Yes. Erythroblast fetus. Erythroblast fetus is where erythroblast fetalis you want. Erythroblast fetalis is very simple. If the father, if the father is Rh positive, mother is Rh negative, okay, what will happen if the child born, okay, if the child born is Rh positive, then the mother's body will recognize this as a foreign particle. But first pregnancy will be okay until there is some problem, okay. Second pregnancy will be a problem. Why? Because the mother would have come across this Rh positive during the delivery time, right? Some mixing up of blood would have happened. So during the second delivery time, if the baby is Rh positive, then the baby will end up getting jaundice or anemia. So we will give Rogam vaccine, okay, Rogam, who will remove the Rh positive from the body or the antibodies that is made against it. Clear? Yes, understood? Okay, now we will go to AIDS, okay, AIDS, acquired immunodeficiency syndrome, which is caused by HIV, human immunodeficiency syndrome, okay. Now students understand, retrovirus, that is your virus who has the capability, okay, capability to convert RNA to DNA. Why? To manipulate the host. Okay, very smart. To manipulate the host, it understood that I have to become the DNA. So it has something called as reverse transcriptase. RT, reverse transcriptase. I already told you reverse transcription. So this retrovirus will have the viral content. It will release the viral RNA into the cell, and this viral DNA will be produced by the reverse transcriptase. After it forms viral DNA, what it will do? It will get into the host genome and it will try to manipulate our genome. It will try to manipulate. It will try to manipulate our genome, and okay, and then what it will do, students, it will get into our central dogma, like how we talk about a central dogma. It will change everything. That is, it will tell viral RNA is produced by the infected cell. So our body will start making viral RNA instead of the host RNA. Why? Because this fellow got in and it manipulated our genome. Now it will form new viruses are produced, and finally you will make many copies of the HIV virus. Now you should understand one thing, macrophages are the factory of HIV. Are the factory of HIV. Understood? They are the factory of HIV. That means they will be producing more and more and more and more virus. More and more and more virus. And but when it comes to when they affect the T cells, okay, when they affect the T cells, students understand, T cells it will kill. It will kill the T cells because it knows that T cells is going to help with the immunity. So we have to reduce the immunity. All of you should understand by now that a person with AIDS, a person will not die because of AIDS, a person will die because of opportunistic diseases. That means what? It will wait for an opportunity. This disease will be waiting for an opportunity to enter inside. That is, if they kill the T cells, that means our immunity will automatically be less, because this is related to cell mediated immunity. Automatically immunity will become less. That time even typhoid comes, no. Even if typhoid comes, that person will end up dying. Okay, even typhoid comes, the person will end up getting affected. Understood? That is called as opportunistic disease. Okay, clear? They'll definitely have weakness and tiredness, all of that will be there. But understand that a person from HIV positive to AIDS positive is a far story. Right, HIV positive a person can be, but AIDS he'll only become after 5, 10, 15 years of a span. Now during this time, this person will go through many infectious diseases. Okay, clear? Yes? Okay. Okay, now cancer students. Cancer is an abnormal, uncontrolled division. Why I am going fast with human health is because in that video, human health and diseases is clearly given. So if you have any doubts, you can watch that video. Okay. Cancer is an abnormal, uncontrolled multiplication of cells resulting in the formation of tumor. Students, just give me 1 minute. Let me just sit for a minute. My back is hurting. Okay, 1 minute. Just 1 minute. Yes, ask the doubt. Jai Ganesh. Students, after this one kutti break we'll take, okay? Yes. Okay. Now students, cancer is an abnormal uncontrolled multiplication, okay? Abnormal uncontrolled multiplication of cells resulting in the formation of tumor. Tumor means what? A mass of cells. Tumor can also be called as neoplast, okay? Neoplast. Normal cells show a contact inhibition. Contact inhibition means what? Okay, that is, let's say I am standing here. Let's say, one more person is standing here. Now, when I touch this person, I'll know that, okay, I have to stop. Okay, my God, no place. But these cells, cancer cells, will not have that ability. Cancer cells have lost the ability of contact inhibition. That means even if they touch another cell, or even if they come in close contact with another cell, they will still keep multiplying and growing. They will continue their division. Okay. So normal cells shows contact inhibition, but these cells do not show contact inhibition because they have this uncontrolled growth property. Okay, clear? Yes. Yes, okay. Thank you. Yes, I will take a kuti break after this, okay. Yes, okay. Now types of tumor students, benign tumor, okay, benign tumor and malignant tumor. Now benign tumor means what? Benign tumor is confined to one location, okay. Benign tumor means it is confined to one location. That is, it is not going to move further. It is like, okay, I am growing here, I will only grow here, okay. It's not all the time it is cancerous. It's not necessary that all the time benign tumor becomes cancerous. They will be here, okay, fine, with a surgery, or we can remove it easily. But malignant tumor is not like that. It will be here, it will go here, from here it will go here, from here, it will spread everywhere, okay. So, and most of the times, no, majority of the times they are cancerous and painful, okay. So, they do not spread to other parts. They do not spread to other parts, clear? Now, can cause little damage, okay, little damage. Now, students, malignant tumor, mass of proliferating cells, that is neoplasty or tumor cells, that grow rapidly, invade and damage the surrounding normal tissues due to active division and growth. They starve normal cells by competing for nutrients. They also do one more very bad thing. They do one more very bad thing, okay, that is, students, not only they grow extremely, let's say this is the cells, okay, this is the cells. After growing next to the cells, they try to fight with the other cells and they try to take the nutrients from this cell. Understood? Understood? They try to take nutrients from this cell. Yes, Ashwika. Gopika ma'am, you called. Yes, I am Gopika ma'am. Cancer, most of the time it will come back, student. like it will recur. Okay, recurring is usually there in cancer. That is one dangerous thing about cancer. Okay. Radiotherapy also, chemotherapy also, if you do, sometimes cancer can recur because of this malignant tumor, because these people will be spread everywhere. Okay, depending on the stage also. Now, like if you have initial stage, if you are in the first stage, most of the time it will be cured, completely cured, but slight chances of recurring is there. Okay. No, only malignant tumor grows by metastasis. Okay, metastasis is shown by this fellow. That's why this fellow is dangerous. Okay, metastasis is shown by this fellow. Clear? Yes. Now, cells lodged from tumors reach other sites via blood where they form the new tumor. This is called as metastasis. Clear? Benign tumor is confined to one place, malignant tumor grows everywhere. Okay? Yes. Now, in case of leukemia, biopsy and histopathology studies blood and bone marrow test for increased cell counts. That is, students, if you want to check if the person has cancer, you can go for biopsy or histopathology. That is, you take a tissue and you study about the tissue. Okay. Now, radiography is done, which all of you know, use of X-rays. The CT scan also is done. That is again to just understand the 3D image of it. Okay. MRI is done. MRI is the most safe one because it is magnetic resonance. Others are all exposure to X-rays. Exposure to X-rays a lot also can cause recurring cancer. Clear? Yes. Now, use of magnetic fields and non-ionizing radiation to detect the pathological and physiological changes. Now, students, use of antibiotics against the cancer cause specific antigen, but again, this is just to increase the lifespan. It's not like a complete cure. There's no medicine, as you know. Molecular biology technique, that is to detect the cancer related genes. Such individuals should avoid carcinogens. Okay. Now again, physical biological carcinogens, chemical carcinogens are there, like your ionizing rays, X-rays, UV rays, gamma rays are very problematic. At the same time, smoke, tobacco also is problematic. Now let's say in our body itself we have something called as proto-oncogenes. Proto-oncogenes are inactive, but the moment they are exposed to UV rays, X-rays on a long run, this proto-oncogenes becomes oncogenes and they become cancerous. Clear to all of you? Yes or no? Clear? Clear? Any doubts? Please ask me. Any doubts? Please ask me. Now we'll go for a break, students, and then sir will come with ecology and my video will go live on the channel. Okay, my video will go live on the channel. So let's take a break. His back is going to break. Okay.
Students, sound check, sound check. Get up. Sound check, students, sound check. I'm audible to all of you. I'm audible to all of you. Sound check. Students, the question practice right, which was supposed to be 6 p.m. I think 6 p.m. is going to be before I finish the chapter also. So what we will do is we'll do the entire question practice at 8 p.m. In 8 p.m. we will be giving you the most important questions. So if you know the theory, just look at the question, you'll get an idea of what kind of questions you'll get. Just read those questions and you're good to go. Okay. Now students, let's start with the ecology. I'll tell you what exactly you need to study in entire ecology. You don't have to learn the entire chapter here. I'll tell you exactly where they can ask you 2-mark question and the 5-mark question here. Okay, students, let's go. Let me just keep my phone aside. First of all, organism and population, what they can ask you. Yeah. Now here they can ask you something called, if they ask you what is the birth rate and death rate, right? There's a small concept of birth rate and death rate. They can ask a 2-mark numerical here. Okay, they can ask you a 2-mark numerical here. For example, if you look at the NCI diagram, here it talks about death rate and birth rate. Okay, now there's a small formula here. Okay, small formula for birth rate. Birth rate. Birth rate is going to be, anyone tell me what is the birth rate, students? Anyone can tell? Remember the formula I told you recently, very recently I told you? Anyone? Newly added population divided by initial population. That is going to be for birth rate. That is going to be for birth rate. Newly added. From a point to the next point, whatever population is increased, divided by the initial population. Now here it tells you, see here. Here is an example. In a pond, there were 250 lotus last year. Through reproduction, 8 new plants are added, taking the current population to 28. We calculate the birth rate. Students, what is the birth rate? 8 new plants are added. So newly added is going to be 8 divided by initial population. Initial population is 20. So 8 divided by 20 you need to do. That's all. That's all, students. So newly added divided by initial population. Now what about death rate? What about death rate? If they ask you a question, death rate is nothing but number of death divided by initial population. Initial population. See the example here. Okay. If 4 individuals in a laboratory population of 40 fruit flies died during a specified time interval, say a week, the death rate in the population during that period is, number of death is how much? 4 are death. Out of total is 40. So 4 divided by 40, that is your 0.1. So one small formula you need to remember here for the birth rate and death rate. Simple formulas are given here. Birth rate and death rate given right here. Okay. Now, apart from this, students, they can ask you the ecological pyramids, right? Age pyramids. So there are 3 types of pyramids here. The first one is called as expanding. An expanding pyramid here, students. Can anyone tell the shape of this? Look at the shape. This is a pyramid shape or a triangular shape. So expanding here, expanding. Okay. Now here if you notice, students, pre-reproductive is more than reproductive and it is more than post-reproductive. Post-reproductive here in this case. And such a type of expanding population, increase in population is mainly observed in your developing countries. Can anyone give me an example of developing countries, students, quickly in the chat? Found in your developing countries here. Developing countries, this type of population group. Now, the second type is called as stable, right? In stable population, you notice a bell-shaped curve. And in this case, students, in this case, your pre-reproductive is equal to reproductive, and both of them are more than post-reproductive here. And such type of population is observed in your developed countries such as USA, any European countries also. Any of your European countries also. Okay, USA or European countries. Now, the last one. Last one is called as your declining population here. And you can see a urn shape here. Urn shape here. This is called as urn shape. Declining population. It is mainly found in the war countries. In the war countries such as, you know, Germany, or it could be Ukraine, it could be your Palestine, and all war countries, and also in case of Japan. Also in the case of Japan. That is the declining population, stable population, expanding population. This much all of you should know. This much all of you should know. More than this, they'll not ask you here. Okay. Next concept is called as your population dynamics. What is population dynamics? For example, there is a population. A population can increase or decrease. Now, what can cause a population to increase and what can cause the population to decrease? That is very important. For example, for this is a population. Birth can increase, that is natality. Death can decrease, that is mortality. It can decrease. Immigration can increase while emigration can decrease. That's all you need to do. Okay. Yes, exactly. Today morning in my paid batch I asked this question. Students, let me tell you, this question I asked in the morning. Many students could not answer this. Many could not answer this. This was the question I asked you. Declining population, what do you see? Okay. Ecology, no one wants to study ecology, no. Keep forgetting ecology. Now students, now there are two types of growth models here. There are two types of growth models which are present here. Okay. Two types of growth model. Now for example, let's say, whose birthday is coming, students? Anyone's birthday is nearby? Anyone's birthday is nearby, students? Anyone's birthday is nearby? No one, huh? No one? Just tie my lace. Don't want to fall randomly. Anyone's birthday is near? Okay. Let's take my birthday is only very near. Okay. Now what happens in your birthday? Right, Jesus, don't do this. Let's say if my birthday is very near, I go tell my dad that, Dad, I want some money. My dad is like, take unlimited money. How much you want? Full entire treasure box you empty of. Unlimited money. In such case, what happens is, when we have unlimited resources, you take a small example. Okay. I have a very large 100 kilometers farm. In that 100 kilometers farm, I have around 20 rabbits. 20 rabbits. Now within 5 years, if I give unlimited food and unlimited space to the 20 rabbits, will the population increase or decrease, students? Mine is next month only. Will the population increase or decrease, students? Will the population increase or decrease? If rabbits have unlimited time, unlimited, like let's 5 years, unlimited space and unlimited food, the population will increase drastically. It keep on increase because no one is there to check on the rabbits. No one is eating the rabbits. Rabbits are present on their own. There are no predators here. Such a type of growth where there is no capital, there is no one to stop the population. That is called as exponential growth, like a rocket is going. See, rocket. Nicely it is going like a rocket here. That is called as exponential growth. Exponential. No one is checking. Keeps on like a rocket is going. Exponential growth. Here the equation is going to be dn by dt is equal to Rn. What is R, students, here? R is going to be intrinsic rate of natural increase. Okay. dn is equal to Rn. Now students, so in reality is it possible to have unlimited space and unlimited food? Not possible. There are 200 rabbits are there, right? By the time it becomes 40, 10 will be eaten by someone. One lion will come and eat the 10. Okay. So what that type of growth where resources are very limited, space is very limited. Such type of growth is called as logistic growth. This is called as logistic growth. Okay. There is no lion there is no lion to come and eat also. No lion to come and eat also. But is it possible that 20 rabbit population without unlimited food, can they become 2000? Can they become 20,000? Not possible. There is a limit to how much a population can increase. I'll give you one more example. Okay. One more example. Let's take, students, how many of you are present in your house? How many people are present in your house? Let's say 4 people are present in your house. I have 4 people only. Okay. My mother makes all the food in small cooker. Now imagine randomly, without even inviting, whole family, entire relatives comes to my house. Now tell me students, is it possible that my mother can make the food for everyone in that small cooker? Is it possible? No. There is a capacity that how much a cooker can make the food. It can make the food for 4 people, but that is the limit to it. The limit here, there is a limit here also. In an ecosystem, students, there is a limit how much our environment can tolerate the population growth. If the population, that is called as K. See, a population can increase, that is called as initial lag phase here, lag. Then there is complete acceleration here. Then we have the acceleration here, and then we have asymptote here. It is stopping it. The population is becoming very saturated here. So initial lag, log acceleration, asymptote. Such type of growth is called as logistic growth when there is a cap here. The cap is called as the K, also called as the carrying capacity. Limited resources, limited space, limited people. It cannot increase. Even in nature also. Okay. What will happen if the population increases? If population increases, there could be natural calamities, earthquake, tsunami, corona, something can happen and population can decrease again. Okay. Logistic, exponential. Logistic with a limited space, limited food. Exponential with unlimited food, unlimited everything. Logistic done, carrying capacity K done. What is our intrinsic rate of natural increase? Exponential, logistic. Formula is very important. Formula is going to be very important here. Students, again, I'm telling you, this R value right here, right? This R value can vary. What will vary, sir? So, students, let me tell you one small example. All of you preparing for CBSE or NEET, right? Now, I can teach the whole day. I can give you all the possible notes. Even if I give you the paper today, there are some students who don't want to study. They're like, I'm too lazy for this. I can't do this. I can't do that. Excuse, always excuse. Okay. So even though you have all the resources, you will still not study. Okay. So this R value is like that. R value is like that means even in the ideal situation, there are some type of organisms who will still not reproduce. Okay, see, if you see the R value of floor beetle, it is around 0.12, so very high growth. At the same time, Norway rat is 0.012, that is very low birth rate. So R value can change even in ideal conditions. Okay. Roshini, human population is still logistic. If human population was going to be exponential, you will not have space to stand also anywhere. The reason there are many people dying, why? Because a lot of people are dying because there is no food for them. Yes, right. There are a lot of people who are dying on a daily basis who do not have food. Right. So it is not exponential, it is logistic. It is going to be logistic only. Okay. Clear? All of you want to kill humans now. Nice, students. Done? Now here you can see there is a exponential equation also. They can ask you question from here also. Okay. Exponential. Nt equals n naught e to the power of rt. What is e? Base of natural logarithm. Students, again I am telling you, this you have to study on your own, because if I teach you this entire concept of commensalism, examples of your predation, examples of your parasitism, commensalism, you can study on this one. Nothing difficult there. Remove the examples of commensalism, examples of mutualism, good to go. Nothing they'll ask you more than that. Examples you can remember on your own. Sure. What is commensalism? Predation is given here. This is very important. This can be asked as a 1 mark question even in a NEET examination. Now, for example, students, someone is trying to attack an animal. Will the animal run away or will the animal stay there? You tell me. Quickly. When animal is coming and when let's say one tiger is coming to eat you only, will you run away or will you stand there? you will run away. Tiger is coming, very difficult. So humans, right, or the animals can run away. They can run away at least. Yes, they can run away. Yeah. They can run away very easily, right? But what about plants? Do they have opportunity to run away? They cannot. So plants have to be very smart. They cannot run away, so they can become strong and stay at the same place. That is called as adaptation. Every single plant has certain type of adaptations to overcome the situation. For example, calotropis. You might have seen calotropis growing everywhere in the backyard. Even behind my house, there is a calotropis tree. No cow will come and eat the calotropis. Why? Because even the cow knows from trial and error. Okay, the cow knows that if I eat this calotropis, I am gone case full. Okay, why? Because calotropis has certain type of toxin called as cardiac glycoside. Okay, very important. In amensalism, I'll tell you again. In amensalism, nothing. Don't study amensalism. Don't study amensalism. There is nothing. Learn predation. Very important. Why predation is important? They'll ask you question. Predators are prudent. What is the meaning of that? Meaning is that very simple meaning. Predators are also sustainable. Now, for example, let's say you went to shopping market. And you got a lot of food. You got chips packet, you got biscuit, you got ice cream. Will you finish all of that in one day only? You'll plan nicely. If you're like me, you'll eat in one day only. But if you're not like me, you'll plan. Okay, I'm like my sister plans. My sister, she plans so much, I will eat first week chips, second week biscuits. What do? By the second week, I eat all the biscuits. She's looking at me now. So that happens in my house, right? So even in the case of nature also, the tiger plans. Like, wait a minute. If I eat all the deer today only, there will be nothing left for next month. They will plan nicely. Deer will plan nicely, right? So let's say today I will eat 10 deers or 2 deers. After 1 week, I will eat 2 more. In this month, my target is going to be 10 deers. Why? Because by the next 6 months, someone must have reproduced and I'll get unlimited food glitch, right? So they think like that. Right. That is called as prudent. They are prudent. Predators are prudent in nature. Prudent. Okay. That's a famous question there. Apart from the students, they can ask you examples. Sea anemone and clownfish example. Mango and orchid example. Right. The egret and the cow buffalo example. That you have to remember all of your own. Okay. What is Gauss competitive exclusion principle? Students, I'll teach you this very well. What is Gauss competitive exclusion principle? In my house, what happens? I'll tell you again. There is one chocolate. One chocolate is coming to my house. One chocolate. Now, both of me and my sister from one room, we will look at the chocolate. As soon as chocolate is on the table, both of us will run. Now, whoever is faster will get the chocolate. Whoever is slow will not get the chocolate. I am fast, I get the chocolate. So I will take the chocolate and I run away. So same thing happens in the nature also. Nature also, what happens when the two species are competing with each other for the same resource, the chocolate. Whichever is superior, faster, stronger, sharper, okay, me. So I will get the chocolate and I'll run away. In the nature also, the animal will get the food and run away. The one which is there now, which is not getting anything, the inferior one, it will perish eventually. That is your Gauss competitive exclusion principle. Now, students. Now, what happens I'll tell you what happens in reality now. Okay. In reality, what happens? As soon as I get the chocolate, what will happen? My sister will start crying. Looking at that, my mother will come. What is the problem? I'll tell mommy, I am faster. I took the chocolate. I got it. This is mine now. Mommy will hit me on the head and she'll tell, you please. Just because you're fast, you got the chocolate. You cannot be like that. Now what do? Mom will take the chocolate. She will divide it into half with a scale. Half piece she will get, half piece I will get. That concept is called as resource partitioning. It is a 2-mark question for all of you. Okay, that is called as 2-mark question. It's a 2-mark question. That is called as resource partitioning. You do resource partitioning there. Yeah. Yeah, see, resource partitioning is here. That's also that's a very easy. Ecology is all about connecting things. Okay. Parasites, all of you know. Endo means inside, ecto means outside. Students, I wanted to ask all the NEET students, let me know in the comment section if you want every chapter to be done in 1 hour. What do you say? What do you do, students? If you do you want it? I'm planning to make a small mini series where I teach you every single chapter in 1 hour, and this 1 hour concept will include all the minor points, this PPT itself. Okay. If you notice here, students, if you notice here, I have highlighted every single important point which can be asked in a NEET examination also here. Okay. The commensalism I have given example here. Mutualism I have given example here. Mycorrhizal association of higher roots. Right. There is something called as Mediterranean orchid or employs sexual deceit to get pollinated by the species of bee. What happens here, students? Here what happens is next level scam is happening here. Okay. This is an orchid. Okay. This is an orchid. Orchid's one petal will look like a female honeybee. The male is like, oh ho, some beautiful female is sitting there. Like, oh ho, one beautiful female is sitting there. But the poor bee doesn't know. It is not a female, it is a petal. It will go nicely, come and sit, finish the business, it will go back again. And he has no clue what happened. No clue what happened. Full scam. Full next level catfishing is going on here. Okay. Now, when the same bee goes everywhere else, pollination keeps on happening. That is called sexual deceit. The male bee doesn't even know, no brain cell, zero brain cell, it is running. Okay. So it keeps on going everywhere, pollination keeps on happening. Plant is happy, pollination is happening. Yeah. This is called resource partitioning, students. Resource partitioning example is here. Now, what about ecosystem, students? Ecosystem, very simple, nothing is there. You need to know the concept of stratification. What is stratification? They can ask you for 2 marks, they can ask you 2 marks question, stratification. What is stratification, students? Right here. This is called as stratification. What is stratification, students? Vertical distribution of species. See, herbs, shrubs, trees. Vertical distribution is happening. Herbs, shrub and trees. That is called as stratification. Now, there is something called primary productivity. Definitions given here, read it once. Productivity, here they can ask you what is GPP and NPP. Under primary productivity, what is GPP, what is NPP? GPP is the amount of organic matter produced by the plants. Plants are producing it. But are they giving it to us? Plant is like, it's mine. Why will I give it to you? Plant is like, it's mine only, I will only use it myself. Yeah. So it is used by plants. So plants will use the GPP, that is minus respiration, and it will give you NPP. This is called as net primary productivity. This can be eaten by us. We can eat NPP, not GPP. Friends, this is a famous 5-mark question for all the board students. Please learn this today only. Do not go for tomorrow morning. It's a famous 5-mark question. I'm telling you now also. I still remember it came in my board examination also. Please learn this. Right, all the steps in decomposition, all the way from the fragmentation, leaching, catabolism, humidification, mineralization. Every single step you need to know. You have to know this at any cost. Okay. What is PAR? Photosynthetically active radiation. That is only 2 to 10% of it is taken by the plants. This also you should know. Properties of humus. Please learn this. Properties of humus. I'll give you the PDF. Here we have something called as the how the decomposition can be affected. Right? What rate of decomposition, changes in rate of decomposition based on that environment. For example, whenever detritus is rich in lignin and chitin, it will be very slow. Decomposition will be very slow. If the detritus is rich in water soluble substances like sugar and nitrogen, it is going to be fast. If it has warm temperature, if there is warm temperature and moist environment, decomposition is going to be fast again. But what if it is cold temperature, low temperature? It is going to be low again. Decomposition rate will be very slow. Okay. What if there is no oxygen? If there is no oxygen, again it is very slow. If there is oxygen, it is fast. That's all you need to know. 3 different pyramids. You can learn on this own. Here they can ask you question. Here they can ask you question. Pyramid of change in pyramids. For example, they can ask you when the pyramid of biomass is inverted. It says here, the pyramid of biomass in C is generally inverted. Very important line. Even pyramid of number can be inverted in a big tree example. In a big tree. It is given here. See this. If you were to count the number of insects feeding on a big tree, let's take, I have one big tree here. This is one tree, pyramid of number. One tree, on this one tree, there are multiple, let's say 100 insects are there. It will be more obviously, 100 insects. Okay. Pyramid of would be obtained. Now estimate the number of small birds depending upon the insects. Now, there are some small birds which are present on this insect also. Let's say I have 50 small birds. This is insect. One tree. So do you see pattern here? We get almost inverted. We get inverted pyramid in the case of number also. Yes. Pyramid of biomass. It is inverted. Why, students? Pyramid of biomass is inverted here because the biomass of the large fish is more and the phytoplankton is very less. Phytoplankton may be more in number, but the life cycle of phytoplankton is so less that it doesn't really give out a biomass there. Okay, doesn't give biomass. Period of energy, it can never be inverted. In biodiversity conservation, students, I'll tell you what exactly you need to study. This they will not ask you, this they will ask you. They can ask you the species area relationship. Here they can ask you this entire equation here. But apart from that, the actual question which they can ask you is this evil quadrant. That is causes of biodiversity loss. This question can always come. Here, I will highlight what you need to remember here. First one is called as habitat loss and fragmentation. Most important cause of biodiversity extinction. Then, here you have to give the example of Amazon rainforest. The Amazon rainforest example you have to give. After that, students, after that, why is it being cut down? It is cut down for cultivation of soya bean, right? Or converted for grasslands for raising beef cattle. Okay. And what is the result of it? This is the result of fragmentation. Many species are becoming, since the area is becoming fragment, many species are migrating or they are dying here. Now, overexploitation, what do you need to remember? In overexploitation, you have to mainly remember are these two examples here. Stellar sea cow and passenger pigeon. Mainly. Okay. That is, instead of using the animals or the plants just for the same need, they're making it greed out of it. They're overexploiting it. For example, how you overexploit your TV time. When no one is looking at you, no one is taking care of you, keep watching TV, keep eating random things. Yeah. Now, alien species invasion, that is multiple things coming into our different countries or different areas, they're coming to our area. Okay, you might have noticed this, right? In school time, what is to happen when A section person is coming to B section? How do you act? Or different section person is coming, A section boy or A section girl comes to your house, into your class. You're like alien person is coming, different section person is coming. Okay, so that is called as alien species invasion. It can happen at the country level. Now, sure, you have to remember the examples again. Nile perch example you have to remember. Extinction of 200 species of ciliate fishes. Parthenium lantana camera, water hyacinth, that is Eichhornia, and the African catfish, Clarus. This example you have to remember. Last is called as co-extinction, very simple. In co-extinction, if there are two species which are interconnected to each other, if one is dying, one more also is dying. Simple example, let's take a fig and a wasp. This fig is in a mutualistic relationship with the wasp. If the fig plant is dying, the wasp also might die. Like if the fig becomes extinct, the wasp also might become extinct because they are interconnected to each other. Like how when your friend, you didn't study anything for the examination, you went and sat in the examination depending on your friend. But friend is like, sorry bro, I can't show you today. Or the friend is like, bro, I didn't study. So friend is also failing, you are also failing because you were depending on the friend. No, the friend also failed, you also failed. Both are gone. And this also can be asked as a question. That is the summits. Two different summits are present here. Okay, the historic events, one is your Earth Summit and the second one is called as the Convention of Sustainable Development 2002, Johannesburg one. Okay, apart from the students, in this chapter they can ask you in situ and ex situ. What exactly happens in situ? What exactly happens in ex situ? Please learn that. It's an easy 3-mark question. Okay. And revert proper hypothesis I have given here. Revert proper hypothesis given by Paul Ehrlich. I think students, I think that's the end of it today. We spent a lot of time today, right? We will give you some questions with answers. Learn those today, right? Whatever you learned right now, it's more than enough. In the end of it, I told you a few things which you have to revise those, but mainly focus on the questions. So when you see a question, you will know exactly what type of question you can expect and what answer you should write also. Okay, so do that, students. Today's class will come at 8 p.m. So students, do you realize from today 10 a.m. in the morning, we'll be doing class till 8 p.m. So it's almost 12 hours here only in the office, right? So 8 p.m. we'll start the class. Join that class, students. Thank you so much and bye-bye. Biotech ma'am is uploading a video already. Ma'am is uploading a video. Watch the video for biotech. Okay. So thank you so much, students. Bye-bye. Take care all of you. All the best and I'll see you exactly at 8 p.m. Go eat something, right? Chill for some time, read something and I'll see you. Okay. Bye-bye, students.